Boy, tava a little thing here, right? Right, right. Tell me about you coming through. Audio coming through, nice. All right. Yeah, sorry about that. first time actually using this platform. I mean, well, for streaming. So, um, well, what we doing today? Well, I can't tell you when this will end. I just kind of doing what I'm doing. Um, this is for, but just letting all you know, this for bio unit 1, right? So this for tomorrow. How are you feeling about that? Well, I think unit 1, the paper 1, tomorrow. Let us get some stuff open here. It working? Alright. It probably have a little latency thing going on. Nice. Well, how are you feeling? How are you feeling about tomorrow? Why not Zoom it? I don't know how much people Zoom could hold. And like if you're doing Zoom, you have to pay for it. Like if it's a large amount of people. Probably in the future, I might do a next one of this tomorrow. So I will see how Zoom working tomorrow. I say I'll try out YouTube today and see what Zoom tomorrow. Actually, the easiest one for me is Facebook, but nobody's be on Facebook. You see, Facebook is for old people like me. Alright. So assuming all they want to go through paper ones, but I don't know if all they want to go through paper twos as well. Well, they tell me. I have the resources for both. I could try them other platforms for tomorrow. Because I'll doing it today and tomorrow. I'm doing it for CSEC too, but all I know no. I mean all they done right CSEC. So by then I might actually figure it out. But it could be too late for all you. P1s now, alright. Well, let me upload something there. Alright. So all I just bear with me for a little bit. I say I still new to these things. I mean accustomed to other platforms. Me I know what the young people and them let's be on. Alright, that's now, eh? P1, I seen plenty people playing P1, so right. Alright, so all you're supposed to be seeing a paper 1 come up there. Just confirm for me now. I say it's the first time doing it here, so we just want to make sure everything good, you know what I mean? Seeing it nice. Okay. Alright. So we're gonna go through as much as we could then. And tomorrow we'll go through the P2. Alright. Shucks, just now, eh? The Alright, the next thing I just want to test here is see if desktop audio coming across. It actually not terribly important, but alright. It blurry? Hold on. Alright, that's not taking this off. But this man come back out here, wait. I know how the thing up. Create new. That's not it. Look, it's better to show my whole screen on none, yes? Because I'm trying to do the window capture thing. And all kind of other thing coming up. Alright, just let me share my whole thing. Yeah, try adjusting the resolution. I on an Ethernet connection here, so I don't know if it's my side. I told you. Well, if we go through some years of paper ones. Alright, so let me look at one. Oh, I wanted to see if my desktop audio coming across. Let's see if I can add that. I could have watched a YouTube video. Hey. I doing this for free, you know. Yeah, but here free thing not good and good thing not free, you know. There's a free thing. If I was going to make some money on it, I would have learned the whole thing inside out. There's just me I home and doing nothing. So feel free to drop in, drop out. Nah, man. Alright, look at this first one. Which of the following best describes the properties of water? You can answer any chance for you. So we have latent heat of vaporization. The tick means a high and the X means a low. So you have latent heat of vaporization, specific heat capacity, and surface tension. And let me do a little zoom up here, so I think those who seen it blurry, it might be a little easier for you. And so what do you feel this one is? So I'm gonna tell you, so it's D, yeah. So just to explain, so all I remember from physics, latent heat of vaporization is the amount of heat that's required to change a liquid into a gas, or 1 kilogram of a liquid into a gas. So water actually takes about I think it's 225,000 or something like that joules per kilogram. So that's pretty high. So specific heat capacity is also high. May I follow remember the actual number, how much joules per kilogram Kelvin, specific heat capacity, and the surface tension is pretty high. This is because there's also bonds between the water molecules. And this also explains, like if you see like water fall on a leaf. If you see water fall on a leaf, you know it is kind of bead, and it does that, and very few liquids exactly do that. So it shows that the surface tension is high. Shania, if I could go through paper twos too if you want to know. There's the majority of people here still want to do paper ones. But I could go through I'll go through paper twos as well. But still we get some paper ones out the way. Alright, let me look at the second one here. So the second question here shows two isomers of glucose. So an isomer is think of it like a different rearrangement of it. So all you should know the names. So what is be ascribed to these types of two glucoses that you're seeing here? I'm gonna do paper two as well. So we'll get some paper ones out the way. Yeah. So where's call these two here? So there's glucoses, but there's isomers, right? So there's kind of like rearrangements. So nice. So we have alpha and beta, right? So by the way, with something that all you have to learn how to do is for the paper two anyways to draw glucose. And usually what you learn to draw is alpha glucose. And well, my students, me know how much of them here know, but My students, I remember when let's see my hey, my mouse stopped working and I can go in no store and get a new mouse, yes? Well the mouse halfway working. But surely, yeah. So you're supposed to know how to draw glucose and sucrose, right? This is 2012, Simone. This is 2012. But I don't have a Jan and keep, so June. Yeah, so right? So this is something that's come up a little bit is how to draw is how to draw glucose. So if we start with let's see, so we know it has 6 carbons, right? It has 6 carbons in a glucose, and the last one is over here, is outside here. And all I remember how is label it 1, 2, 3, 4, 5, 6. That's 6 carbons. So put in the chart there was the name for 6 carbon sugar, no? So we have 6 carbons and an oxygen, and all I know, well, carbons have 4 bonds. And I remember it like this. So I remember ho. Started with carbon one anyway. So ho. You have to remember to make sure that you have 6 bonds. A hexose, nice. So I remember ho. Oh, shucks. So it not too bad to draw. So you could draw this one as HOOH. I'll just draw it as OH. But I mean when you're drawing it bonding now, you could draw it with HO. And well, this last C here and this CH2OH. This aldehyde group. So what's something all you should learn, just practice a few times. Well, I guess not for tomorrow, but for Friday, is learn how to draw a glucose molecule. They have sucrose in the syllabus, but that'll really come. Is mostly glucoses come, and you should know the difference between alpha and beta. So the beta this H and this OH here swapped. So and I can label C1, C2, C3, blah blah blah. So just remember ho. And you just need to make sure that it have 4 things attached to each carbon. Nice. So coming back here now, what people in here? I me no nothing no. How's this now? How is him check that? Participants. It have a good bit. I was expecting to have like 2 people. I expecting to have like one. All right. Let me go ahead. So let me look at this question again. We go work at a faster pace. I just kind of abide in time a little bit so that if other people come in, they're gonna be too lost. All right. Which of the following statements is true about all you hear for me? I say I just doing this. So it's true for both isomers. So which one all you think this one is? Question 2. Which of the following is true? So all you remember that this one is alpha and all you remember this one is beta. So which one they're looking correct? This is question 2. So we have the alpha glucose and beta glucose. Which one it is? We have both contribute to starch, both contribute to cellulose. One contributes to cellulose, two contributes to starch, one contributes to yeah. So that's D. Yeah. Sorry about that. Serious? What is in then? So right. So one contributes to the structure of starch, two contributes to the structure of glucose. So cellulose. Sorry, cellulose. If I remember how. Nah. So if anybody, you can put it in the chat. Cellulose. So cellulose is not like a string of beta glucose. So it's not like a straight string of beta glucose is. How the arrangement is, or let put it in the chat. How the arrangement is for cellulose when you have the beta glucoses. So it don't be like one after the other like straight. It made out of beta glucose, but it not like connected as a, how I could say, boy, it's not like it's the same thing. So it's linear, but each, right. So each one in each alternating one is inverted. So it's not like it's the same thing over and over and over again. So it's not like, how that's how amylose is like the same thing over and over. Right. So it have a 180-degree flip. All right. Wait. Let's tell me to zoom out. Hold on. Right. So it flipped. Okay. So the answer for this one is D. No, we're trying nothing. We're trying to get it right. All right. Look at this one. Phospholipids. So you talk about a phospholipid bilayer, right? Yes. So it have 1,4 glycosidic bonds and have 1,6 glycosidic bonds. Phospholipids will affect the function of which are the following cell structures. So you have the ER, centrioles, mitochondria, and nucleolus. So there's only two of these that correct. So the one that we're looking for here, we're looking for things that have a double membrane, right? So things that form like a full organelle. So you're looking for things that have the double with inner and outer membrane. So which ones here that do have that? So centrioles. Oh, they asked me if I could save me, and if I save in this, I don't know how to save nothing. I just start up the thing, and I say people go come. Yes, there's the massive amount of H bonds that makes cellulose sturdy. So the two here you're looking for the double is the endoplasmic reticulum, right? And the mitochondrion. Check now. Hold on. Oh, I now see. I now checking my OBS there, and I see any madness not happening. I see any madness not happening. I see. I now see any madness. The thing when I put it now, the thing went way. Right. Tell me some improvement now. Tell me if the thing was zoom right up. It wasn't my computer. It wasn't like the computer. It was like when I put the display right. Yeah. The thing went right in. All right, all they should be seeing better now. Yeah, all they should be seeing better. Anyway, so it's 1 and 3. The ER and the mitochondrion. So what's that? That's B. The so centrioles don't really have. They not really comprise of phospholipids. They like have microtubules and all kind of other things, but they don't have that double membrane. So remember, phospholipids have the amphiphilic, that have the hydrophilic and the hydrophobic part. So centrioles, if I remember, centrioles really only found in animals, and they help with cell division and things like that. But they're actually pretty simple structures that they do have double membranes. The nucleolus, if you remember, is like a wrapping, a very dense wrapping of chromatin within the nucleus, and that do have a double membrane, so it wouldn't have phospholipids. So anyway, so the answer here will be 1 and 3. Right. So nice. So we good then. We good. So the thing working better now. All right, so you look at this one here. Samples of a solution containing carbohydrates are treated as follows. So you have Benedict's reagent have no color change. Yeah, the ER have one. The ER, remember that they have, I don't know how to pronounce that, but the ER have cisternae, like these little folds. And I think the folds only there because they have phospholipids. Yeah, but the endosymbiotic theory would prove it for mitochondria and chloroplast, yeah, but the ER is part of it. It wouldn't be as complex as the other two, but it have nothing else there that is match. Anyway, yes, they hold the ribosomes in those little cisternae. Yeah, yeah, like the mitochondria, except you just call them cristae in the mitochondria. All right, so Benedict's reagent by itself had no color change, right? So we know that this here, well, this test for reducing sugar, right? Or they forget my, right? So this test for reducing sugar, and so this one here, this means that absent. Um, HCl Na2CO3 and Benedict's root. So this proof positive. So this would be non-reducing sugar, right? And amylase added, and then Benedict's solution. So amylase would digest the starch, right? And then that starch would break down into maltose, well, some kind of non-reducing sugar. So which of the following carbohydrates are most likely present in this solution? So what you all think there? Shucks, this YouTube thing close. Yeah. Well, the so I understand like a lot of people leaning towards B, but really the answer here will be A. And so let me explain why. This first one here, this non-reducing sugar here is sucrose, right? And when they say in the solution, if they add amylase to the solution, that amylase, remember, enzymes are very specific and it would only break down starch. So they're talking about the solution after. They're talking about the solution before. So before they add that amylase. So the best thing that would work here is sucrose and starch. The so anything with glucose would be wrong because glucose, if it's Benedict reagent alone, it had a turn brick red, and yeah, but in that case it didn't have a color change. So it'll be sucrose and starch. So four will be A. Oh, by the way, in case some people said they didn't see this one, so two would be D, right? One contributes to starch and two contributes to cellulose. I don't know. I don't know how is record alive. I don't know nothing about these kind of things. I didn't watch no video, how to do this, I'll tell you, the chances is me, The chances are I don't know. So if that a jump in. Assuming that when the live when it is end, it have a thing to say save or at a record now or something. I say I do it on YouTube because they say that at the end. They say at the end the thing might save. But I don't know exactly how you save it. Alright, let me look at 5. Alright, that's now Yeah, maltose is a reducing sugar. No, maltose is reducing. Even though it's a disaccharide. Right, so five would be B, which are the following is represented by the structure. So we're looking for these bonds here. these carbon nitrogen bonds is what we're looking for. So here we have sucrose is not reducing here. Maltose, even though it's a disaccharide, as I said, is still reducing. So this here is a tripeptide. These are three amino acids linked together by peptide bonds. We're looking for these here. So this is also something that you should know how to draw. So we know that, right, so that's a tripeptide. So when we draw in this here, so right, so you should know how to draw amino acid, right? So now we have the amino group here. We have the not NH3, NH2. Then we have COH, H, and all you could tell me what is usually go in this last thing here? Right. So you'd have your R group here, your residue. So this would be your R group. And what I would usually be is, so something else you have to know for paper too is that they sometimes have a question where they would say what is the group, or explain how amino acids are differentiated from each other or something like that, right? And it easy. You just have to say, well, all of them have these carboxyl amino and H groups, right? Now each one has an R group, but the R groups are different. And I don't know if anybody remember off the top of the head how many different amino acids are there, and what does actually make those amino acids different is the R group. Sometimes it's be as something is be as simple as just a hydrogen. Sometimes it would be a long string with all kind of nitrogen rings and all kind of thing. Right, so it's a 20. So it's the R group that differentiates them. The other things are constant. So anyway, there's a tripeptide. Right, so 20 amino acids. Oh, what's the name of the process that is turn? Oh, the other 21st one? May I keep up now? What is the thing that is make the proteins? What's the name of the process that's make the proteins? That's take the um the genetic code and turn it into proteins. What's the name of that process? No, not condensation. Condensation is when the bonds forming. But what's the name of the process that is actually take the genetic information and right? So translation. So translation is what has caused that. But the actual formation of the bonds is due to condensation, I think. So all right, that's now just give me a little minute here. Just checking something. I just replying to our text here. All right. Don't worry, small thing. All right, which of the following best describe the structure of collagen? So a triple helix, a alpha helix, a double helix, or a beta helix? Yes. And the name of the RNA that deposits, sorry about that, I know this guy anyway. Right, so it's a triple helix. And I'm not sure I'll picture here. Yes, transfer RNA is what takes, each one has an amino acid attachment site, and that is what forms, that's what deposits the amino acids onto the ribosomes and they link together by peptide bonds. Yes, covalent as well. All right. So a blurry, hopefully it's not my end. Right. So if you look here, so here we have the peptidases. Don't worry about that for now. The amino acids, right? So that just as I do in this multiple choice, I kind of going through these things, so I hope all you don't mind, or if all you just want to go straight multiple choice, multiple choice. The amino acids, right? So there's a repeating sequence. We know that a repeating sequence, and that is unlike, so that's why it's called a fibrous protein. There's a is insoluble and it's a repeating sequence of amino acids, like over and over and over again. And what happens there is that there are three different helices. Well, three different strands of these, and they intertwine with hydrogen bonds. So they form like little cross links and thing. So here what we have is three different strands intertwine, and what's hold them together, these hydrogen bonds. This here makes them good for structure, kind of like cellulose. You know, cellulose is like it has like a lot of structural integrity. Um, so they're not like enzymes. So like enzymes, yeah. So you would have many of these collagen molecules that would further wrap around to form collagen fibers. Yeah. So if we think about all you should remember that it have four levels organization in proteins. So we have primary, secondary, tertiary, and quaternary. And usually it's not so much collagen that is asked you to describe. Yeah, kind of like root. That's a good example. And they ask you to describe the four levels using a protein. And usually the protein is not collagen. The protein is hemoglobin. When we get there, we get there. So yeah, it's a triple helix. Alright, carbohydrates are transported in plants mainly in the form of what do you feel for 7? 49 are good, boy. I say I was expecting 3. Yeah, carbohydrates are nice, sucrose. What's the name of the vessel that's transported? What's the name of the vessel? Yes, it's less reactive than glucose. So it wouldn't be glucose because glucose is too reactive. Phloem, nice. So the phloem, through translocation, would transport that sucrose from a source to a sink. So like if sucrose have to connect, like roots can't undergo photosynthesis, right? Because roots do have chloroplasts and they're underground, so that they can't undergo the light dependent reaction for photosynthesis. So when the sucrose, when the sugars are stored, they store as starch, and the starch is way too big. It's not really what I could say is not too non motile. It's not too mobile, and so an enzyme called invertase breaks it down into sucrose, transports it. I like to equate it as this. Um, imagine like you're going down the road, like you're transporting real gold or real money or something, right? And if you're transporting it as glucose, it's kind of like you're transporting it in a little normal car. Well, by the time it had to go from A to B, everybody teeth out all the glucose and all the glucose get absorbed. But if you carry it in an armored vehicle, amalgamated or something, that's kind of like sucrose. It less reactive, it less liable to be absorbed by other things. Yeah, the biochem is a real thing. Will this be live be posted at hey, I go try. I go try my best. I go try. Alright, look at item 8. Item 8 refers to the following diagram representing the structure of an animal cell as seen under electron microscope. Which of the follower which of the following structures present in a cell would not be seen in a bacterial cell? Alright, we don't really need to see the diagram, but which of the following here you wouldn't find in a bacterial cell? Ribosome, cytoplasm, cell membrane, or nuclear membrane? Yeah, nuclear membrane. Well, that's because, well, bacteria are prokaryotes, right? And they don't have a nucleus. What they have in placement of a nucleus? Anybody know? Oh, our next thing that ribosomes, right? Bacterial cells, they do have ribosomes, but they're not the same as ribosomes in a eukaryotic cell. What's different? Yeah, plasmids, they'll be in the form of plasmids, and the whole region is be called this the let me put it there. They'll be called the nucleoid region, also plasmid, circular DNA. Yeah, so prokaryotes, prokaryotes have 70S ribosomes and eukaryotes have 80S. 70S, just think of that as smaller. It is really a unit. It stands for Svedberg unit, the S. It really, think of it like, is sort of like density and how fast something would sink in a medium. So it literally just saying it less dense and it's smaller. So it's MTS. All right. Okay, so here we have a transverse section of a plant. Um, so the area labeled X, X is what you're seeing here, right here. What is this? Is this a cell, a tissue, an organ, or a system? I do lessons now, boy. I actually done as a teacher. I's not a teacher anymore. So lessons out. Yeah, there's a tissue. So you know tissues are a group of cells, right? A group of cells that interact to perform the same thing. By the way, which tissue is that? What tissue is that? Yeah, I's not a teacher, I know. So yeah. All right, yeah. Nah. That one is not the xylem. Yeah, that's the phloem. So how I remember like this little X, this kind of like a cross, like a X. That is the xylem. That is the xylem. And we boy here there's the phloem. Anyway, good thing they asked that. Okay, so um here we have a diagram and we're showing it goes from a high, basically down a concentration gradient, a high to a low. And we see it passing through these things here. So the figure above would best represent which are the following: diffusion, exocytosis, active transport, or facilitated diffusion. Well, one time we could knock out active transport, right? Because that's from low to high. Yeah. So facilitated diffusion. The what was this? Was this? Was a was that thing? Was this here? Yeah. Well, not channel protein so much, nah. Have a next word fit. You see, the thing about channel proteins is that they kind of, yeah, carrier proteins is the better thing because channel proteins, they kind of like tubes, they kind of like tunnels, but carrier proteins, they have to connect with the molecules, they have to interact with them to open up and let some through, and some might flow the other way, all kind of thing. So, by the way, so why is it not why is it so technically diffusion correct, eh? But it's a best demonstrates. So if it was diffusion, like if it was simple diffusion, it would be passing through the bilayer itself. Yeah, like probably simple. I don't really know what that is, nah? Right. But this would be simple diffusion. That would be simple. If it passing through if it's small enough to pass through the phospholipid bilayer. And somebody did say it there, but the one that is transport water is called an aquaporin. The special type of channel protein that's transport water. I didn't say what this is, but you're assuming it's you're assuming it's glucose or something. I don't something not too big to pass through the phospholipid bilayer. Yeah, so that's 10. Okay, so 11, when a plant cell is placed in a solution with a less, hey, there's always a chain up thing, eh? A less negative water potential than its cell sap, the cell becomes more what? So if something less negative, that mean it have a higher or lower water potential. All right, I got answer that water question just now. Right. So less negative mean higher. So when a plant cell is placed in a solution with a higher water potential than its cell sap, right? Which one here go work then? So if we have a cell, right? So if you have a cell and the plant cell is placed in a higher in a solution with a higher water potential, yeah. So it could be A. Let me explain this there. All right. So let me answer that question there with the water passing through the bilayer. Let me answer that one day. What really happening here? Why? Oh no, I'm trying to get all this crap on here at the same time. All right. Let me answer this bilayer one. Um, so water molecule shaped, right? Water molecules have a oxygen, and they have a hydrogen and a hydrogen, and they have a dipole structure, right? So they have a plus and a minus here, and right. So what happening there is that even though the fatty acid tail is hydrophobic, um, which means that they repel water, right? Water itself is very, very small, like as a molecule, real small. And the most of them wouldn't be able to go through. But I would imagine that if it have enough water, if it have so much of it, imagine like you're trying to get through a gate, not a gate. You're just trying to get through. You're trying to go for your vaccine, and there's a big open thing, and all the security guard and them is the hydrophobic and them now. They're pushing you use the water. They're pushing you. No, they can't come for no vaccine now. But it can have a few that will get through. Right? So even though it have these forces pushing it away because it's so small, and you would have it's not a large amount, but some will still go through. The majority of them would pass through aquaporin, which is a water filled channel protein. But if you have enough of it, some of it will still get through. Despite it being hydrophobic, it's because it's so small. But if it's like larger molecules that that water-based molecules, they won't be able to get through. But water itself, some of it still could. It's confusing, but in the end, just say just say water, just go through the aquaporins to even mix up yourself. All right. So, all right, let me explain this one. So it's A. Let me explain here. So they say here we have a plant cell. They say there's a plant cell, and we have a less negative, right, water potential outside here, which means that we have a more negative one inside here. Less negative just means it just mean higher. They just want to chain you up. More negative mean lower. And well, we know with osmosis is take water molecules and make it flow from higher to lower. So what gonna happen here? The thing going to get turgid. They're going to fill with water and they're going to get turgid. Yeah, that's it. It's as simple as that. They're literally just saying if you put something in a higher water potential, what will happen? Yeah? Okay. Which of the following events does not involve the movement of ions or chemicals across cell membranes? So we have cell to cell recognition, antibody interaction, chemical stimulation at a synapse, active transport by enzymic pumps. Oh, shucks. Sorry about that. Yeah, more salt inside. Well, more solute. It ought to be salt. Right, does not involve. I see in E. Bulleting. Oh, and there, yeah. Others. I'm trying to see really happening here. Yeah, sorry about that. Right, so it could be A. So movement of ions or molecules across cell membranes don't really involve cell to cell recognition. If all I remember, cell to cell recognition is due to like the attachment of glycoproteins on the proteins, the extrinsic and the intrinsic. Yeah, so cell to cell recognition is everything else here would work because all of these here deal with something moving across. Active transport deals with something moving across. Chemical simulation and synapse, although you don't use do that in unit 2. Um, with neurotransmitter and thing that something moving across. Again, acetylcholine are moving across. Antigen antibody interaction, same thing moving across. Cell to cell recognition is kind of the odd one out here. So 12 is A. That more deals with like recognizing certain things just to eventually see what to do with them, but it doesn't really deal with movement. All right. Here we have a relatively easy one. Pepsin is an enzyme found in the human alimentary system. Which of the following activity curves represent the action of pepsin? Well, we know that. Well, if where the pepsin is, like where is find it? Where is find pepsin? Yeah, yeah, like antigens. When you get your Sinopharm vaccine, then you produce them antigens. You know what I mean? Right. Okay. So the answer is A. But where pepsin is stomach, right? Yeah. So it has an optimum pH of about close to 2, which means it's very acidic. And yeah, you would find pepsin in the very acidic stomach. Um, which one here you all think would represent trypsin, by the way? One, two, three, or four? Trypsin is kind of like pepsin cousin, or pepsin brother. which number do you think would represent? Yeah, trypsin would be like 4. Let's say this would be trypsin, and this one here would be pepsin. Trypsin works in a very slightly alkaline. Anyway, that's not what it asks. Nah, not 3 could be like amylase or something. Trypsin could be a little, that only they do it in so. All right, really going on here, boy. All right, I'm trying to get this to fit. Uh, all right, 14. Item 14 refers to the following graph, which shows a relationship between enzyme concentration and the rate of an enzyme catalyzed reaction. So here we have the rate of reaction, or the V, the velocity, and the enzyme concentration. Based on the diagram above, and look at how the line is, which of the following statements best describes the rate of the enzyme catalyzed reaction? Yeah, trypsin in the pancreatic juices. Things like trypsin and lipase and erepsin and all them other things you probably didn't learn. Yeah, these. So they're directly proportional. That's easy. Kind of question is that? Yeah, so those things there, they help to neutralize the stomach acids because you could get intestinal ulcers and things if too much stomach acid enters the ileum. Um, that and bile. This help do that. Yeah, so anyway, directly proportional. You should know this. Well, you do CSEC. Yeah, if it's a straight line through the origin, thing is a directly proportional relationship. I don't know if I need to explain that. All right, so let me look at 15. Well, 15 shows effect of increasing substrate on the rate of an enzyme catalyzed reaction. And which of the label curves best represents the presence of a competitive inhibitor? So I go explain competitive enzymes is one of the topics. Or they have in all the paper too. Enzymes is one of the topics. We they take the easiest topics in the paper too. From what I see, it was what like biochem. No, what they take out heredity. They take out natural selection. They take out enzymes. They take out membranes. They're all them easy thing. Anyway, so the competitive could be B. Well, check now. Let me explain. So it could be B. The let me explain. I mean, if I come in paper two bag still yeah, natural selection. Yeah, there you go. Natural selection is really the best topic because there's always actually same questions over and over, and that's real tough luck. No. Or everything in paper one, but paper two have a limited number of topics. Hey, I wanted to ask all you too. What going like Chem and thing going right? Did the CXC tell the truth? Like that's really the topics, or they bring other things. They tell the truth. All right. Well, that's good. That's good. Cause I hear. I don't know who doing literature, but I hear literature. That they bring all kind of thing that wasn't there, but that's probably a failure on the literature group. All right. So let me explain this thing. I won't stay too long with it because they're coming. You see here we are enzyme. There's an enzyme. right? That's an enzyme. And we say that there's a substrate, right? There's a substrate, and this here is not a substrate, but is something that have a part that shapes similar to a substrate, right? So there's an imposter, and this is what can happen here. So the substrate will fit. Yeah, well, animal and plant reproduction is the whole of module 3. Oh, shucks, just now, eh? For transparent. Right. So a substrate could fit into the enzyme and that would create an enzyme substrate complex. And you get some condensation reactions or some hydrolysis reactions occurring, wherever, right? And this substrate will break up into products. And that is what you want to happen. You want the enzyme to work on the substrate. But how this is that it have a specific little space that these substrates would fit. And that's called an active site, right? Or activation site. And so all you know, like an enzyme is a protein. It have a name to describe that type of protein. So with G was the name of that type of protein. It's soluble, it's specific, it not made out of non-repeating units, it not really used for structure. They're very highly involved in metabolic activities. What type of protein are talking about? Yeah, globular. Right, so globular proteins. So the globular. And what was happening here is that this little active site here, you would have a bunch of R groups that would kind of be jutting out. And remember, each R group specific, like each R group different. So in the amino acid chain, you would have a bunch of R groups sticking out, right? And so you might have alanine, leucine, cysteine, all kind of different things. You have many different types of combinations, and that would bond to this and break it down. Now this could happen now if you have a molecule that is very similar or has a part or a chain of amino acids or whatever that's very similar. This could end up happening, right? We imposter could end up, you don't call it an imposter, right? Just going in. And that could end up going in here. And well, sometimes what could happen is nothing could happen sometimes, right? Sometimes it could end up binding temporarily or partially, and it's kind of like, well, it will go in and come out, but what it's really doing is just wasting time. It wouldn't really have a reaction like the other molecules. It it might be temporary. It might be partially, and it will lodge partially. But the point is that it interrupted the whole thing. And all of a sudden now what we have is these two competing for the same space. It's kind of like musical chairs. So you might have many different types of substrates. Watch now. Me I say nothing, eh? Watch now. You see a R word you see there? So me I say number there, eh? So do I mark papers? CSEC. But I am aware of how CAPE is marked. Also, I'm not a teacher. Again, I'm not, right? So yeah, so anyway, this will come in here. But I know the syllabus inside out. The So when it comes in there, we say it's competitive. It's a competitive inhibitor because it's just taking up space. They competing for the same space. Now, what could happen now is that an enzyme it do just have one site. one binding site. They might have another binding site here, and let me outline it. In blue. So this site here, all you know the name of this? Or you know the name of this site here? Right, so this is called an allosteric site. And allosteric sites literally mean, allo means other, right? So it means other. So it just means like another site where this binding can happen. And what could end up happening here is that this could be was who's like a man who's Tifa Gilway, like Kyle or something, be Kyle, right? And so what Kyle going to do, right? Is he going to gain here? And what gonna happen now is that usually what will happen is that when an enzyme has to link up with a substrate or with a has a bind to it, it actually changes its 3D shape a little bit. It have a name for that. Something something. So it's kind of like when you put on a glove and the glove fits around your hand. Same thing when the substrate goes in the ends and the enzyme will sort of fit around the anneal. Well, the substrate is anneal. So what would happen here? Induced fit, right? So what would happen here is that this whole enzyme going to change shape, right? So the whole thing going to try to wrap around this thing, and all of a sudden now the whole thing might actually change shape a little bit. So all of a sudden now we boy anneal again, right? So this is a non-competitive inhibitor because they're not competing for the same site, and this is usually permanent when this happens. So trying to get with again, and all of a sudden now the enzyme is a big waste all of a sudden because it's not going to perform what it's supposed to do. It's not going to take substrates and turn them into products. It's not going to allow the binding of the substrate supposed to do. So if amylase, it's kind of like when it gets denatured. When amylase gets denatured, the starch can fit in it again to break it down into maltose. So I'll give you an example of this. It have this group of chemicals called organophosphates, right? Organophosphates, and what those do is that they end your bop and your death and all your bug sprays, and when you spray the bugs on them, the what happens is that those chemicals, those organophosphates and the pesticides, they actually occupy the enzymes in the insects. So what happens is that the insects cannot undergo normal metabolic reactions. So their body would basically go into a seizure. They wouldn't be able to respire. They wouldn't be able to move properly and have like motor function, like voluntary motor function. That's when you spray something, it is wriggling up. So that is a non-competitive. Anyway, but all you again tested on that. But I just saying in case it come up in a multiple choice now. All right. So let me look at, well, all you know how does this go? It is a 45 questions in a paper 1 and 15 per module, right? So we go bounce up plenty repeat something as we go in. Item 16 refers to the following diagram showing a strand of DNA, a segment of a strand of DNA. And so we know DNA is a double helix, and one of them does run 5 prime to 3 prime. Let's think about it. 5 prime to 3 prime, and the next one is run 3 prime to 5 prime. Give me one word that is described that. Give me one word that is described that. By the way, the 5 and the 3 is referred to the carbon number. So there's like carbon 5 and carbon, right? Right, anti-parallel. Nice. Okay, so this is the base X. What we had is cytosine. All right, so why we saying cytosine then? So if you look at T is bond with, or if you remember, I'll put it in the chat. If you remember, apple under the tree, right? So you're adenine to uracil, well that's an RNA, or adenine to thymine, and that's in DNA. And you remember car in the garage. Yeah, car in the garage. So cytosine to guanine. So this is 2012. Right, so the base X. So this thymine, this here would be an adenine, right? So we know that is not adenine. We know it's not uracil because uracil is a RNA thing. A tree grows corn. Hey, that's a good one. I don't know if a tree does really grow corn though. Anyway, so we have two types of nucleic acids. We have purines and we have pyrimidines, right? So purines have two rings, and pyrimidines have one ring, right? One nitrogen ring. So what we're looking at here, because this box is bigger, this is a purine. Right? Yes, so pure as gold and cut the pie. So this is how you remember which one is a purine. Purines are adenine and guanine, and pyrimidines, right, are cytosine and thymine. So what we're looking for here is which one is the purine, and the one that will be the purine there would be guanine. Wait, let me make sure. Pure is here. Wait, is it? Let me make sure. So thymine to adenine, guanine. Is guanine or cytosine, right? Pure as gold. No. The one to know is X. Right, so the one to know is the pyrimidine. I so studying to figure this one out. Is cytosine. Yeah, sorry. This is guanine. This could be guanine. And this could be cytosine. Yeah. So the base X is cytosine. Hey, let me know if all you good with our explanation. Let me know if you're good in it. I find that one is real. That one is real to you know. Like to be honest, that question is a real thing. Let me type an explanation. There's 2012, 2016. Three bonds indicate I type in something. Three bonds indicate that it is a guanine cytosine bonding, right? Because that are triple bonds. The purine or double ring is portrayed as a larger box, which is guanine. This means that the smaller box, which is a pyrimidine, has to be cytosine. That question is real poor, but the answer is cytosine. I thought, give me a little second here. I just had to go and do something for a minute. Right, Ollie. Sorry about that. Okay. I get to just look a little clearer. Oh, and here. Tell me, tell me if everything showing there, like, it's a little better. Good? All right. All right. Look at this one here. Which of the following gives the correct sequence of events in the replication of DNA? So bonds between complementary bases break, bonds form, opposite strands separate, blah blah blah. Which one all you feel is the sequence? If you could figure out, well, the last one is two, right? The last one is two, but which one it's starting with when we replicate in DNA? Which one it's starting with? What's the first thing that has happened? Right, one is first, right? So that knockout C and D. That knockout C and D. So it's either A or B. So let me see what looking like the next stage. Right, so then three. So the answer is A. There's a D. So bonds between complementary bases break. So that's the hydrogen bonds we're talking about. Right? Them bonds will break. That will split the two strands of the DNA. And we'll go through this in more detail tomorrow. The opposite strands will separate. That's kind of like the template strand and the leading strand and the lagging strand. Those two would separate. Then free nucleotides that just floating around in the nucleus or floating around in the cytoplasm or whatever. Those are going to pair up. So A is going to pair with G, A is going to pair with T, sorry, and G is going to pair with C. Remember talking about replication of DNA. So when they're talking about transcription, adenine is going to pair with what? With transcription. When they form a messenger RNA molecule. Nah, I don't to know the measles thing and measles and style experiment. It has helped to know how it is, but it's you have it not in the syllabus. You're still good. And something else I was going to ask, but when I forget what I was going to say yes, but anyway, so it's one. Oh, right. Replication of DNA. What is called that in the cell cycle? It have a name. So you know it have telophase and anaphase. And the what is called the stage where replication of DNA is occur. It's basically 95% of the cell cycle. What is called that phase? Interphase, yeah. So interphase is where you have most of this replication occurring. So more on that later, probably tomorrow. Alright, so item 18 refers to the following diagram showing part of a DNA molecule. And here we have, well, basically C with G, we have a sequence here, right? C, A, A, T, T, G, blah, blah, blah. And they say which of the following correctly identifies the sequence of bases of the mRNA strand produced during, during transcription. So what we want to look for here is to see, we want to look at this strand here. We want to look at the leading strand, well, the 5' to 3' strand. And we want to see here, yes, pure maths unit 1. Nah, boy. Right. Yeah. Sorry. I know this. So which one here would form the RNA? Which one here will you think? So what would it really is, you just have to see what would pair with CAA. Was the complementary RNA to CAA? Yes, the ribosome reserve, but it formed as from the 5' to 3'. GUU. Right. So there's the answer. Right. So it wouldn't be GTT because it's an RNA not forming, and the RNA don't have thymine. So it has uracil. So the answer here could be GUUAAC. Right. So you're looking at different codons: GUU, AAC, UUG, and GGG. No, you would read the right side one when I want to make double sure of that, right? But I believe, let me see, transcription occurs. Let me make sure. Let me make absolutely sure. In what direction? Along which strand? DNA only one strand. I'm just making absolutely sure. If it's leading or lagging. Nah, it's 5 to 3. It's 5 to 3. As I say. So it's still It still is. When is when it is when it being read by the ribosome is a different direction. Yeah. Yeah, buddy, I zoom out. Hold on. Hold on, hold on. I'm trying to get us to. Alright. What I see and what you're seeing is like two different things. Anyway. Alright, there you go to the next one. Which of the following statements describes the difference between RNA and DNA? Right, so what they have here? They have RNA is double strand, DNA is single strand. What that's wrong? RNA has uracil as one of its organic bases, while DNA does not. RNA has four different nitrogenous bases, DNA has five. RNA is found only in the cytoplasm, DNA is found only in nucleus. So which one all you feel it is? Well, it's B, right? This real thing, why? RNA and DNA both have four. It's just that thymine is be replacing well, uracil is replaced thymine for RNA. DNA is found not only in the nucleus, DNA is found all over. You just find DNA all in the mitochondria and ER and all kind of thing. It's just, it could be different arrangements of the DNA, but yeah, you find DNA everywhere, not just in nucleus. Because it could be in different forms. It could be in chromatin, heterochromatin, euchromatin. And remember, like when mitosis and thing occurs, you just get nuclear membrane, all kind of thing breaking down. So yeah. Which two of the following features are evident during mitosis? Okay, so what do you think? Two daughter nuclei containing identical sets of chromosomes. So this is correct. Right? It don't have any crossing over, so this is wrong. Four daughter, this is wrong. Right? Homologous chromosomes do not associate. Sorry, so it's one and four. The so it's B. So tomorrow I'll go through mitosis again, but before paper two stuff. But basically during mitosis, they independently they are aligned independently along the metaphase plate. That's the middle of the cell. They don't pair up or anything during mitosis. They don't cross over. They don't touch. They don't form no chiasmata. Nothing. So they just kind of align independently and get pulled apart. So it's one and four. Right. Homologous, when they say homologous chromosomes don't associate, as I say, they are aligned independently. It's not like they paired up. So like, let me show you a quick thing here. Let me show you a quick thing. Let me add something here. Add. I feel like I get any hang of this thing. Alright, so let me explain. So mitosis, like you said, is the cell, and there's we chromatids, right? Or we chromosomes, I should say. So what happening here is you're gonna end up with some crossing over with your chromosomes. So during mitosis, the chromosomes align independently in the equator of the cell, right? So they're like separate. And meiosis, you have crossing over or synapse, called synapsis occurs. So you'll have basically DNA being exchanged at these points here. So in the question here, what the question was asking, right? So what the question was asking was which are the foreign evident during mitosis. So they do associated, which means that they do crossover, which is true. 18 and 19, 18 is C, 18 is C, and 19 is B. Yeah, they don't have the crossover, the chiasmata. Yeah. All right. So look, in a rat cell, there are 32 chromatids present at the beginning of prophase of mitosis. How many chromatids would there be in the cell at the beginning of prophase 1 of meiosis? What they would say? Nah. It's not 16. Yeah, it's first prophase 1, eh? It could be it could be the same 32. I don't know where 23 come from. It's the same 32. So prophase, remember, prophase is just the formation of these denser chromosomal structures. So when those chromosomal structures is formed, I know there's a big chain up scene here. This one is a big chain up scene. So let me show you what I mean here. That's now, eh? Where this thing they way. Right. So this one is a big chain up scene. Prophase 2 would be 64, yeah. No, prophase 2, sorry, would be half. That would be the haploid number, would be 16. Right. Let me explain something for you. This, when all you see a chromosome, right? Right. So it would have double up during interphase. So this is one chromosome, but two chromatids. Yeah? So that's one chromosome. The chromosome is the whole structure. They made out of two strands there. This, when you see it like this, like when they pull apart, when it's like this is one chromosome, one chromatid. It's still called a chromosome. So when they pull apart. So this is why I go through this in more detail tomorrow because we're doing multiple choice now. But when they get pulled apart, so like when this here, get pulled apart, right? And you end up with this, like when they get pulled apart during telophase. So when they get pulled apart, well, not telophase, not anaphase. When they get pulled apart, this is two chromosomes, two chromatids, two chromosomes. Brain bus there, or So this is one. When usually it's seed formed during prophase, this is one chromosome. It replicated during interphase. This is why it double up like this, like a butterfly now. So it double up. But so it's made out of two chromatids. When they get pulled apart, right? We have one chromatid each, or two chromatid, but this is now two chromosomes. So you're basically counting each structure. So when they count the structure, that is there what we'll call a chromosome. Yeah, I know it's confusing, but I guess tomorrow we'll go through it a little more. Right now we're just doing this multi choice. All right, now you continue. Uh, Move on to other things. But I say, my last year teaching I go do this because I know this year was real hard. This pandemic year, and CXC real dread to still have exams. So I say I have all the resources. I go pull a thing. Whoever want to come and study, cause like what all they doing today anyway? What they doing today? Me, studying. You might as well I'm here. All right. Look at this one. Which of the following statements best explains a null hypothesis? All right. So a null hypothesis is usually what you do, like when you're doing a statistical test, like a chi-square. And so a null hypothesis is where you're gonna assume which one, that there's no significant difference, that there's a significant difference that is not testable. Well, we knock out C and D one time, it's not that, right? So it's either A or B. So which one it is? So the null hypothesis states that there's what? Right, there's no significant difference between the observed and expected data. So all you do chi-square is really to tell so that you'll get a probability value that would be able to tell you if there's really a correlation, like a significant correlation or not. You don't have chi-square in your paper too, so I'm not really gonna go through it. And they don't really ask chi-square questions in paper one. This is like the only one that's really come up. Hypothesis which states that there's no significant difference. All right, let's look at this one here. When a cross between a pure breeding plant with green round seeds and a pure breeding plant with yellow wrinkle seeds, all of the F1 progeny. Sarah Ann, I doing as much papers as I until I get tired. I'll probably do this whole day, almost whole day. I'll probably do it into the night. So bring all your food and thing. When a cross was made between a pure breeding plant with green round, blah blah blah. Right, which of the following ratios shows results of a test cross of the F1 generation? Okay, so yeah, so your answer is A here. And let me explain it. What we have here is two purebred. Purebred means like homozygous for the traits, right? So I could do the Punnett square anything here, but basically what we would have is when we have a purebred breeding with our next purebred, right? We will get the ratio that we'll get the same number for each trait. So we would have basically, if you break down the ratio, it'll be one of each. So the other one that is significant is when you have a 9 to 3 to 3 to 1 ratio, right? Which is not the answer for this one. But when what case we would have if it was a 9 to 3 to 3 to 1 ratio? How would we describe the two P plants or the traits, however, if it was a 9 to 3 to 3 to 1? Yeah, it gotta be hetero for both. Those who joined, I doing it. I doing it in like any order, honestly. I start with 2012 because that's probably the earliest year we could go back. That yeah, yeah, it'll be heterozygous for both traits. So if we have let me just say RRWW, right? Pure breeding with yellow wrinkle seed, right? Going this like common R WW, common W. That would yield this here. But if we get in this ratio down here, which is the other important one, it would be like this. Both heterozygous. Well, I don't know if any only write in paper three. You only have to worry about this if only write in paper three because this not any topics. Alright, anyway, so is it. Alright. Oh god, well, okay, so sometimes some of these questions be kind of ridiculous, you know. Like, look at all this information here, all this information for 1 mark. Well, actually, it's 2 marks because each multiple choice question treated as 2 marks. But let me look at it. Um, Here we have restriction enzyme. So just to give you an example, just an idea of what is a restriction enzyme. A restriction enzyme is also called an endonuclease. Is basically, imagine it like genetic scissors. What they could do is that they could cut out piece of a plasmid, and but when they cut it out, they a lot of the time they don't cut it out. They just kind of cut it out like this. So imagine that we have a plasmid, and let me let's see. So imagine that you have a plasmid, and there's a plasmid, right? That's a plasmid, and when they cut it out, it will leave like little uneven ends. So it might look like this. If all I could imagine that. So the thing might, the piece that they cut out, it might look like that. So this this is the plasmid. There's the plasmid, which is like circular DNA, and this is the DNA cut with the restriction enzyme. And so you notice how we have little pieces jutting out. So these little pieces jutting out, these could form hydrogen bonds, right? These could form hydrogen bonds here. Hey, Ola, give me a second here. My phone ringing, right? Give me a second. Hello, good afternoon. Yes. Yes, sir. So, how are you going? Right. Mhm. You can call them. I don't know if they open right now, but you call them and see now. They would have all that information. They just usually have all that in files. Mm. Mm. Call them now. Yeah, see if you can contact them and ask them. And let me find one for you. All right. So All right, take down this number, right? 6M2 8797. Yeah, it's not guaranteed that you'll get through to them today though, but still try. Nice, okay. Well, good luck with getting through that. I know them think could be a hassle. Nah, nah, call the school and ask them. Yeah. All right, no problem. Take care. Sorry about that, y'all. So, where we dey? Right. So, as I say, what is call this part here? Like these parts as jot out. It have a name. Anybody? Sticky end, right? So, what a restriction enzyme is, as I say, they kind of like genetic scissors, and they could cut out like piece of they could cut out a sequence from a plasmid. Now, I actually find restriction enzymes is like this big genius thing that I don't even like that you really have to know your bio to discover something like this because just the idea. But imagine now, like you know how scissors now, each restriction enzyme different, so which means that each one could cut out a different sequence, right? And they ascribe names to these different restriction enzymes. So here we have BAM H1, Echo R1, HPA1, and HPA2. And what this is showing here are the different sequences that it could cut out. Don't worry, you don't have to memorize any of these sequences. So like if you look at a simple one, HPA1 cuts out CCGG, right? And GGCC, it actually doesn't leave a sticky end here, which makes it a little difficult to attach. But what we're looking for here is to see which of these enzymes here, these restriction enzymes, could actually cut from this sequence here. So it's basically matching. We matching the sequences here. So I'm trying. Let me see if I can get this to fit. Nice. Okay, so look at this is more like you don't need to understand how you don't need to understand how restriction enzyme works. Is basically just matching sequences. So all you look and see which ones matching there. Which sequences are you seeing that could fit that could cut these? Which restriction enzymes match sequences here? So for example, if you're looking for BAM H1, are you seeing a GGATCC? If no, that means it's not the answer. So it had one that you could definitely see right away. This question actually pretty easy, you know. Which one looking so? Well, if you no, have a little answer. Sir? Yeah, it says D. So if you look here, CCGG GGCC, right? Well, look at it, right? And if we look at this one here, GTTAAC, right? Look right here. That question you need to understand how restriction enzymes work. You just had to see what sequence matching up. All that in for that question. Anyway, but the answer is D. So there's 24, right? So 24 is D. How it is cut? Is an enzyme. An enzyme known to hydrolyze and break bonds and all kind of thing. You need to go beyond that. Right? Enzyme just break a specific number of bonds, so that's how we're doing it. But really, why they exist in the first place is really to to get rid of plasmid bacteria and all kind of thing. So when we have bacteriophages, hemiplasmid and plasmids and bacteriophages and thing now. So the Israeli a tool, a weapon that bacteria use to fight off viruses. All right. If you look at item 25, refers to the following stages used in the process of genetic engineering. All right, so we have a potential difference is applied as a voltage. Cut by restriction enzymes, DNA extracted from the cell, fragments travel to the anode. We have a probe that's added, fragments placed in the well at the end of the gel, autoradiograph. All right, so what really the name of the process that they describe in here is a process that's called electrophoresis. And the way to just, I think once you identify the first step, the rest of it is, yeah, you need to know what's going on. So what do you feel is the first step here? Yeah, the first step would be just to extract the DNA. You have to get the DNA out. And then you cut the DNA using restriction enzymes to get the sequences that you want. Then you put the fragments at the end of the well. So basically what it's doing here is that it going to, if in chemistry, kind of like chromatography, where the colors just kind of separate and go up this chromatography paper, except it's not colors, it's a genetic sequence, and your denser sequences would be at a certain point, and less dense would be at a certain point. That's kind of like how does work. Let me actually show you something like as a result of it. Anyway, so the answer here could be B. Um, there's some open something here for you. Uh, because all they're probably like, what the hell is that way? Let me show you a practical, or at least the result of it. Um, hold on, biology. All right, so if I watch here, recent. All right, so if I watch here, right? Yeah, but it call electrophoresis, and you just end up getting like different bands of DNA. So if all you ever watch Maury, who is the father? The What they would do is that, remember, the baby would have half the mother's DNA and half the father's DNA. So if all you look here, the baby here we are the baby's DNA. Well, we're looking to see, we could look here and see, well, where the baby get the mother's DNA from, right? So the baby get this band of DNA from the mother. It get this DNA from the mother. It get this from the mother. Where's it get from the mother, boy? And I get this from the mother. So we're looking at the other things here. So who's the father then? So all these other things here, the baby would I get from the father. Well, if you is the father there. It is basically like how a paternity test is carried out. There's do this electrophoresis, there's get the DNA from the mother, DNA from the father, and there's do this DNA fingerprinting thing. Yeah, so Adam is the father. Yeah, so this is it. So it's Adam. Now if you look here, well, the baby had to get this band from somebody. So this is either Barry or Adam, right? But if you look at this one, if you look at this one here, only Adam have this. Well, where does this baby get this? Baby can't get it from Cortez or Barry. Them don't have that. And the mother don't have it either. If we also look at what's the next one? If you look at this one here, well, nobody had that except Adam. So that's kind of like the process to describe in there. So what it is do is that there is add a probe. You don't have to know it in detail and thing. So what it is do is that why it has so much potential fathers, things is happening by time stuff. There's plenty waste man and thing out there, that's why. So what's gonna happen here is that they will extract the DNA, they'll cut it with restriction enzymes, like these sequences down to examine. It wouldn't be like the whole genome. Um, then they will place it in little wells, like those little fragments. They'll place like using a dropper, no, like a very like a micro pipette. And then what they'll do is that they'll apply a voltage, and the voltage is going to do is the voltage is going to help to separate those bands. It's a very small voltage. And the next thing that's going to happen is that this voltage is going to separate, and the fragments going to travel from one end to the next end. So it have a cathode and an anode. And then they're going to add a probe. And basically what the probe, think of the probe as like a kind of fluorescence. And it's kind of like, when they go in for X-ray and they tell it to eat this barium. Going for a stomach X-ray, they eat this barium so we could see what's going on. It's kind of like that. It's kind of like a tracer. So, and then the autoradiograph is actually depict the bands. So that's electrophoresis. All right, so look at 26. Okay, so here we have, everybody asks if I could save this live. I don't even know, I don't even know what I'm doing, you know. Or they ask me to do things I don't know how to do. I don't know what I'm doing. I know biology, that's why I know. I don't know about saving thing. Alright, so item 26 refers to the following diagram, which illustrates a form of gene mutation resulting in a change in chromosome structure. Alright, so basically we have C and D being taken out, so all you could guess. You're taking out something, so what it is? C and D gone. They're cut out. They reach. Yeah, that's a deletion. Yeah, so that's a deletion, right? So 26, which form of mutation does it? Right, so it's a deletion. If you're adding something, by the way, what that would be? If you're adding something, like you get extra base, extra nucleotide or something, what that would be? Yeah, that would be an insertion. All right, that easy. All right, so next one. After which of the following forms of gene mutation will a change in codons occur? All right, so when they say a change in codons, right? Imagine like you have these codons here. All right, that's not. Let me come back here. All right, so imagine this. Imagine that there's we sequence here, right? So we have TTT, ATT, GGG, GTT, GTA, whatever, right? This is we sequence here, and each triplet that we have, each triplet here, this is called a codon or a triplet codon, right? And what's going to happen here now is that when the mRNA is produced and then it gets sent to the ribosome, it will get translated into a protein, right? So what will happen here is that imagine now we end up with this. We end up with TTTT, right? ATT, GGG, GTT, GTA. Well, we can't write the thing like that because our codon is only 3. So what will end up happening here is that this could be written like this: TTT, TAT, TGG, GGT, TGT, A whatever, right? So what it was before was this. So what would happen here is that this would remain intact, but this now is different from this. This now is going to form a completely different a different amino acid. How can a substitution occur if the pair, right? Okay, I can answer that just now. Right, so this is going to form a different amino acid, and this too going to form a different amino acid, and this is going to form a different one. So what we have is a difference in how we're going to read the code, because the code is read 3 at a time. But if we add in something or take out something, we are actually changing the frame of the code. So this is called a frame shift mutation. Right? So this is called a frame shift mutation. So basically what they're asking here is which one's a frame shift for 27. Which ones result in a frame shift? What do you think? Basically, what changes the entire reading frame? Which is actually which could result in very, very bad mutations because the entire protein would become warped as a result. The 3D shape it would fail to manifest in the way it should, I should say. So actually, wait, they ask for a frame shift or not? They just ask will a change in codons occur? They didn't ask for a frame shift. So yeah. Addition addition and deletion could be frame shift, but substitution would change the codons too. Yeah, sorry, I misread that. Yeah, so it's 1, 3, and 4. But these two here, these could be frame shift. Substitutions the result in frame shifts. And yes, Kimberly's right. Some of the codes are degenerate, which means that sometimes it could have something swapping out or something substituting, and you could end up with the same you can end up with the same amino acid. Yeah, inversion is usually chromosomal. All right, so look at this one. Giraffes are thought to have evolved to have long necks due to a short supply of food. Selection pressure favored those with long necks who were able to reach enough food to survive. So when natural selection favors one thing more than the other, that is called directional selection. So you imagine like you have long neck giraffes and short neck giraffes, and of course the larger population going to be the long neck ones because they can actually reach for food now. So that's directional. Disruptive. I go through the others because all again that in paper too, right? So directional. Okay, so a possible benefit of producing transgenic plants. Which one will you feel? This one easy. It's like 4 5 thing. 29. All of you. Possible benefit of producing transgenic plants. Improve here. Yeah, like golden rice. Yeah, so like golden rice with the added beta carotene to it. Yeah, that's an easy one. Which of the following plays the most significant role in allopatric speciation? Just to break down the word allopatric for you, allo means different, and patric comes from pater, which means father. So allopatric literally means another fatherland or another land. So which of the following plays the most significant role? That would be geographical isolation. It's literally another land. Was a good example of allopatric speciation, like one that's often studied. If you had to give, well, I mean, paper two, you'd have to give an example. Well, yeah, I have this, but what's a good example of allopatric? So this is where you have a physical space between the two, and usually there's an ocean or a landmass separating it. Yeah, Darwin's finches is a very good one. Our next good one is I don't know if you learn about the abe and the kaibab squirrels. They live on opposite ends of the Grand Canyon. So I mean, literally, there's a large gap separating the two types of squirrels. But Darwin finches is your go-to one. Okay, I'm gonna zoom out here a little bit. Okay, so 31 is they don't have asexual reproduction in the syllabus again. Well, they have it, but it's not like a full topic, but we'll still go through it. Which of the following methods of asexual reproduction is incorrect, either by its definition or its examples? Okay, so we have binary fission, which is one cell, basically one cell split into two. Fragmentation, splitting into several pieces, each of which regrows as like ginger. Budding, producing an all growth from a parent breaks off, and spore formation, producing small lightweight spores. Yes, so this one is wrong. And why it's wrong is because the list hibiscus and flamboyant as your examples, so that's wrong. The definition correct, but hibiscus I think don't produce spores. Um, if they did say like mushrooms or some kind of thing, then yeah, but no, not plants. Also, these don't reproduce asexually; these reproduce sexually. All right, so spore this last one. This is correct, eh? But this is wrong. All right. Item 32 here, bamboo and sugarcane reproduce asexually. Alright, so item 32 refers to the following diagram, which shows some stages of spermatogenesis, the formation of sperm, in a mammal. And here we're going to start with the spermatogonium. These are like the undifferentiated, cells that would eventually become spermatozoa, but they could also become other spermatogonia. So we're looking to see what exactly is happening at E, F, and G. All right, so what type of cell division is occurring at E, F, and G? So we know that by the time it becomes a spermatozoa, it is a haploid cell, right? So it's going to be a haploid cell, and we know that haploid cells are formed due to meiosis. So E is going to be mitosis, right? So these are going to form a lot of different spermatogonia. Mitosis is, remember, is what we're going to use to clone itself, right? And F and G are going to be meiosis 1 and meiosis 2. So I don't know if anybody could name what are these intermediate stages though. Anybody had that one? These intermediate stages. So you have spermatogonia, then we have, what's the next one? Was the ones that lead up to spermatozoa. Spermatogonia is kind of like the male version of oogonia. Again, what they could do is that they could either make more spermatogonia through mitosis, or they could specialize to form other things. So the other things are primary and secondary spermatocytes, and then you have spermatids, and then you have spermatozoa. Eventually, what would happen is that spermatozoa would be formed due to the end of meiosis. Okay. All right, all right, all right, all right, all right. How many people we have in here? Like 10? All right. The following are advantages of two types of pollination. You have no need for another plant, increases vigor, widens the gene pool, no loss of advantageous phenotypes. Which of the above specific to cross pollination? 95 people? There plenty, why? Okay. Hope all are learning. Which of the above are specific to cross pollination? Okay, so cross pollination is when, of course, when pollen goes from one flower to another flower. So it's no need for another plant is self, right? So well, I mean, come now, man. They have one. There's something. I mean, when you're looking at these questions, right? And we know one wrong, right? Because this is self, not cross. And this one have one, this one have one, and this one have one. So the answer is C. 32 Rita is D. D for dog. Alright, so 33 C. Increases vigor and widens gene pools. Cross pollination, you can have a loss of phenotypes because you can have a dilution of a phenotype. Let me say that you have a nice red flower and it ends up crossbreeding with a pink flower, and you don't get a nice red flower again. You could end up getting with some pink spots. You could end up with something that less red. So there's a chance of that happening. The When they say vigor, what vigor means? Vigor is kind of like a next word for fitness. So when you have exchange of DNA, you always have you have a chance to get more variation, genetic variation. More genetic variation is good because you would end up with plants that could be resilient against different types of pests. They could survive in different climates. They could the if it's cross pollination, the plant could end up um, what plants from one environment can interact with another environment. So you get increased fitness as a result. You get a lot of genetic variation. So 2 and 3. Okay, so 34. This one actually shows up a lot. Item 34 refers to the following diagram, which shows the development of the embryo female gamete. So here we have the embryo sac, and the embryo sac. Just to draw this out for you, right? Um, that's now, eh? All right, so just to draw this out for you. So we're gonna have one little starter cell here, right? This we megaspore mother cell. And what's gonna happen here is we're gonna get 4 of these. Right? So you're gonna have 4 of these, but how much of these survive? Well, for females in fact plant and animal systems, they actually spend a lot of energy to produce embryos or embryo sacs or whatever. So you don't want to produce 4 embryo sacs, that's a lot of energy. So what will happen here is that the 3 of these are going to degenerate, right? So these are going to degenerate, and this one here now, this is going to undergo, well, first it's going to grow, right? It's going to grow, and so get bigger, absorb nutrients, and then eventually is going to undergo mitosis. So you're going to end up with it doubling the number of cells each time, and we're going to end up with 8. 1, 2, 3, 4, 5, 6, 7, 8. So where we start off with here, we start off with a diploid cell, right? Which means it has 2n number of chromosomes, right? And what we have here, this is a haploid cell. So this here, all of this here has just one set of chromosomes, right? So each thing here. So here we have 8 haploid cells, okay, in the end. So that means that what we have here is meiosis. That's making this meiosis 1 and 2. And well, this is just development. And here we have mitosis, mitosis, mitosis. And then, we have some specialization and thing that's going to happen. So in the end now, what's going to happen to this one is that this will, this will get specialized, and you're going to end up with a structure that looks, well, that'll be represented like this. We'll end up with 2 central nuclei here. We'll end up with 3 cells, down here. We end up with 3 up here. And that's basically a total of 8. Right? And this is our developed embryo sac. And these here, we just call them the polar nuclei or the central nuclei. Here we have the antipodals or antipodal cells. The word antipodal, by the way, literally means, podal means like foot, like pedal. So it means on the other foot. And here we have these 2 over here. These are called synergids. Well, synergids comes from synergy, not the TV station, but they work together. And what they just do is that they just help out with Gale in the center here. And this is the egg. Right? So this is the egg cell. And so like when when people just go out, or like, and they're going to pick up girls or pick up boys, if you're doing it by yourself, it you just look like real thirsty if you're doing it by yourself. But if you have somebody helping you, kind of guide, now open up the little gates and thing for it, it's a little easier. And it's like a wingman or a wingwoman or whatever. And so the synergids are kind of like that. What the synergid does do is that the synergid does guide the male gamete, secrete like little chemicals and things that would guide the male gamete to the egg cell so it could form a zygote. So when we have the pollen tube coming through here, through the mycopylon, and there's a male gamete here. There's a male gamete. One of them going to fuse with the egg cell, and the next one going to fuse with these polar nuclei here. And they're going to form a 3N nucleus. What is called what are those form? Oh, remember? That triploid fertilized, well, post-fertilization structure. What are they? Yeah, that's the endosperm. And that would be like the food source now. For when it developing into a seed and into a fruit or whatever, like when it starts up, it's like basically endosperm is if all they ever drink coconut water, all they drink level endosperm already. Anyway, so the question here is, the processes occurring at 1, 2, 3, and 4, right? So one is meiosis, right? And 2, we just have growth. 3 and 4, all of this is mitosis, because from these arrows going through here, all this is mitosis. So where are they? What is? Which one had a meiosis, growth? And nuclear fusion. Right, nuclear fusion, because these 2 polar nuclei would end up forming one central cell. So which ones have with meiosis? Right, meiosis, growth, right, so it's A. Yeah, the pericarp is the coconut jelly. In some plants, the androecium matures before the gynoecium. I never think so, boy. Right. Such plants are described as what? Okay, so the androecium is the male part, the gynoecium is the female part. And this is basically what is called a well, what this does is a mechanism that prevents self pollination, right? So if you have the male parts and the female parts not form yet, well, it's unlikely to if they form at different times too, or they form in different seasons or something like that. Then what that would do is there's a cross pollination that promotes cross pollination. So it prevents self fertilization. It's kind of like this. It's kind of like anybody here is go anybody's go Asja that is over school. The boys and the girls that is over school at two different times. They are all to talk to each other. Oh shucks, oh shucks. I forget to move this thing. Wait now. Where the thing going away? Just now, huh? Hold on, eh? I think it closed off by mistake. Hold on, eh? Hold on. The thing closed by mistake. What really going on here, boy? Just now, eh? Check them, yeah. That's now, eh? I just. Give me a minute. All right, that's not. Let us put the window cap today. Give me a second here. Hold on, I just moving all this thing all the way. Move all the way in on them. That's so much thing here. Like a real loose track. Do like Wayne come and see you all you say then. All right, just give me a second. All right, all right. All they supposed to be seeing. All right. Me know what happened there. All right. Let me know if all you seeing. Yeah, yeah. Wayne had the power to give all your grade six in her. Like you can get all A's and thing and you still get six in her. Them thing happened last year. Let me know if the thing showing now. All right, nice. Okay, there you go. So in some plants, right? So I was saying, you know, like Ajda, that there's over the girls' school before the boys' school or something, so that they don't mix now. So imagine like we have two schools, a boys' school and a girls' school, and over the boys' school first. So they say all the boys go home, right? So that basically what's happening here, so that the boys and the girls don't mix or something after school. So what's gonna happen here is that the stamen gonna mature before the carpels, that's like the ovary, I think. And so what we see in here is protandry, well, protandrous plants. Pro means before or front, so it literally just means the males mature before or males mature first, right? Pro means like first or front. The opposite, if it's the gynoecium maturing before the androecium, is called protogeny or protandra or protogynous. Um, Dioecious plants is where you would have separate male and female. The word dioecious means two houses. So it's like Ajda again. Yeah, 34 is it. It's like Ajda again, where you have a boys' school and a girls' school. It's like naps. You have the boys and the girls. So 35, that'll be dioecious. So like when you like or you know example of a dioecious plant, like plants that see you have male ones and you have female ones. Yeah, pawpaw is a good one. Cannabis is one too. You know, you could buy your 4 wheat plant, but that to be one sex. I can't remember if it's male or female. I don't play up in them thing. All right, so you look at this one here. Okay, so here we have uterus. There's like a form 5 question. Which of the following correctly identifies the structures labeled PQRS? All right, so P is a ovary, right? Q is a what? P is ovary. 4 male wheat plants, all right. So P is the ovary, and you'd find that at the end of the oviduct, the fallopian tube. Q is that's the uterus or that's the cervix? And then we have endometrium and myometrium. Anyway, the answer is B, right? Then we have endometrium and myometrium. To give you the difference, so when girls get the period, they grow extra vessels, like the endometrium becomes highly vascularized. You know, you get a lot of blood vessels and blood flow, so that you get a lot of growth in the endometrium. This is so that implantation could occur. Like if a sperm cell meet an egg cell and form a zygote, and you basically get thickening of the endometrium, and this is due to like a number of hormones like estrogen and progesterone, right? And the endometrium is the top, is the surface most layer of it, right? So that's R. And when menstruation occurs, the endometrium becomes devascularized, which means that the a lot of the blood vessels in it constrict. So what really happening is that when girls get in the period, when they're menstruating, is basically the you could think of it like the endometrium strangling itself and closing up and shutting off all the air. It is literally choking itself to death. And what's gonna happen is that the cell's gonna undergo necrosis, well, they die, and they don't have enough energy to hold on anymore, so they shed. They go through the cervix, they go through the vagina, they go into the pad or the tampon or whatever, and that's what happens there. The myometrium is what helps to dislodge the endometrium when menstruation occurs. Myo means muscle. So this is the muscular layer of the uterus. So these muscles contract and relax, and this is what causes the pain. I don't really know the pain of a period. I assume it very annoying, right? Um, I did have a kidney stone, which was the worst thing in the world, and you know the pain is not a nice thing, and it's not. I assume it's not very nice to deal with it once a month. But that is all thanks to the endometrium strangling itself and killing itself, because it basically very disappointed that it didn't have a baby. So it basically choked itself to death. And the myometrium helps to get rid of it. It helps bury the body and toss it out. So myometrium is muscular layer. So 36 is B. 36 is B. The the P is pointing to the ovary, the circular structure. If it's pointed to the tube, it would be it probably be A. But if we look at the diagram, R is the endometrium, the topmost layer. All right, so look at it. In plants, the ovary is best described as the what? Okay, so the ovary. This is a question that comes like all the time. It comes like all the time, right? So remember this one. So almost every year you go bounce up this stupid question. So the ovary is the swollen base of a single carpel, right? Um, the so the tip of a carpel is a stigma, right? The basic unit of a gynosium is a carpel. So a gynosium could have three carpels, for example. So they could have three stigmata and three ovaries. So the best example is the swollen base of a single one. It's not the way I would describe it, but that's how it is. All right, so 37, let's see. All right, 38. Um, Here we have, it looked like an alien, eh? It kind of look like ET. Anyway, so here we have a follicle. Well, not a follicle. We have inside the ovary, we have a depiction of ovarian follicles during different stages of development. And we have one, two, three, four. I'm gonna zoom up a little bit. And here we have to say which option describes the correct order of development. Okay, so we need to start with where it is a primary oocyte, and or is basically just on the very follicular stages. So which stage do you all think we should start with? And which stage would we end with? So when you have a question like this, that is what you really want to go into. Actually, but it's probably more important to ask which one it ends with. So when we have development occurring, right, you're going to start with stage 3, so this is going to be stage 1, right? This is probably like a primary follicle or something, primary oocyte or something. And then the next stage is going to be, you're going to end with 2. So the next stage is going to be this here. This is probably a secondary oocyte. This is the antrum and so on here. And then this is the third stage. This is where we have the rupturing of the follicle and the release of the secondary oocyte. Zoom out. Oh, yeah. Right. So we have the second, then this one here. This is the last stage. This is what's left behind. Anybody remember the name of this thing here? So it's 3, 1, 4, 2. All I remember was the name of this thing here. Now, we have reached corpus albicans yet. That's like the scar. It is leave behind. This thing's still operational as a corpus luteum. Corpus luteum literally means yellow body, right? And it's known for like secreting progesterone and thing. All right, so yeah, luteum. Which of the following statements describes, so in 39, which of the following statements describes the events occurring in asexual reproduction only? All right, so this one is not hard. So when we have asexual reproduction, there we see. This is a Form 5 thing. Offspring would be genetically the same because you have no crossing over DNA, and gametes are not involved. Right? You would have other mechanisms like budding or fragmentation or something like that. Okay, look at this next one here. So the next one is, what is the function of testosterone in spermatogenesis? So what testosterone does, right, is that it promotes sperm production, and it let me see, inhibits the activity of Leydig, inhibits the activity of Sertoli, results. All right, so this wrong, this wrong, this wrong. It will inhibit those things. So the only one here that could work is D. Yeah. It well eventually, like after a long, like indirectly, it would do the Leydig thing, but we are go so far with it. So they say with spermatogenesis, so we can stick to that. Results in the growth and development of spermatogonia and spermatozoa. So yeah, we are going to go into that feedback loop and thing now. We just do any multiple what fit in any multiple choice. Alright, so you look at 41. 41, when will ovulation occur if there's a surge of LH and FSH on day 12 of the cycle? So on day 12 we have this LH surge, right? So when will ovulation occur? Like what y'all think would fit? What y'all think would fit the best? Hopefully everything's showing. All right. So when you just have this LH surge, right? Um, what we want to look to see is if there's a surge on LH and LH. Let's see. Let me just take a close up on this diagram here. Okay. So I think typically when you have that surge, it's not like within 24 hours. It is be a little time after, but I wish the time frames real short here, but like they're real close. They don't like woman could really tell when the period gonna happen, but I believe it's 48. I think is about 2 days. Let me just double check that just to make absolutely sure for you. So I believe is 2 days. LH, because usually why I saying 2 days, right, is because I know that ovulation, if you was to take a 28-day cycle, it would be um at the 14th day. So it's usually be like in the middle of the cycle. So yeah, it could be C. That's a real dumb question. I mean it's not esoteric, it just I don't know. The answer's so close. I mean the only one there that could be wrong is D. But I guess they're talking about typical because honestly it depends on who is the person it's occurring to. Because sometimes it could be within 24 hours. But I think the best answer here is 48 hours. I think they're talking about the average person. Anyway, 42, during the development of a fetus in the womb, the wall of the uterus consists of a layer of muscle and the what? So when you have uterus, you'd have the layer of muscle, that's the myometrium, and then you would have your endometrium. And along the endometrium, you would have the chorionic villi and thing that would form. And in case you can't remember what is the chorion or the chorionic villi, those are the the little finger-like projections, and those are going to connect the maternal and the fetal bloodstreams. And so the maternal bloodstream is going to be the endometrium, and the fetal bloodstream is going to be in the chorion. So you're gonna have like some active transport and facilitated diffusion that are going to take across your glucose and your sodium and things like that. But the uterus, this is specifically uterus, is going to be endometrium because the chorion and the villi are fetal structures, not maternal structures. So looking for maternal, the only maternal one is the endometrium. All right. Here we have a section through a seminiferous tubule, and I'll zoom out a little bit. And here we have FSH acts directly on the cell's label what? Okay, so FSH, just remember this. I'll put it in the chat. Acts on Sertoli cells, and LH acts on Leydig cells. So Sertoli cells are kind of like nurse cells. They help with providing growth to the spermatocytes. They kind of like, like a cow has the little udders underneath that the calves just feed on. They can Sertoli cells are kind of like that. Leydig cells are found in the interstitial parts of the seminiferous tubule. You wouldn't really find them inside the tubule itself. They would be in between the tubules. So the one here that looks like a Sertoli cell would be 3. You could kind of see, you could see here is this big cell here, and you could see, you could see it. You could see like, and I say it's like if you ever see babies suckling on like a calf suckling on a cow's udders or something like, it's kind of like this. It's kind of like these little sperm cells, like the suckling on the Sertoli cell. One interesting thing about the Sertoli cell, by the way, in the future, maybe not too far into the future, when they finally get that male contraceptive pill working, they deduce that it might be that they would deactivate some of these Sertoli cells or make them less efficient or something like that. So that men, they would still produce semen, but they wouldn't produce very effective sperm cells. They produce like sperm cells that's starving and malnourished, and you wouldn't form a baby with it, right? They get pelt out and they can't swim. So no flagellum and thing. So 43 would be C. All right, 44. A birth control method that is used after sexual intercourse is typically referred as a easy one. So there's a morning after pill, right? I don't know how to save a video. If the thing asks me to save, like when it done, I can save it. But I don't know when I'll go watch it. Tomorrow? Is AM or PM tomorrow? Is the exam. A birth control method that is used after, right, is a morning after pill. You do something that I regret, you take your morning after pill. Alright, so if you look at item 45 refers to the following diagram. AM hard luck day, boy. Them AM exams is pressure. Refers to the following diagram, which represents a human embryo developing in the uterus. Alright, so the structure label X is the what? Here we go with CXC, can't make out nothing kind of scene. What is I really looking at in this diagram, right? Here we have a fetus. Here we have the chorion, amniotic sac. This look like the chorionic villi. So what would fit here would be C. Site of exchange between the mother and the fetus. Well, I was saying that since all the P1 tomorrow, I gotta do mostly P1 now. P2, I go do this again tomorrow, you know. More doing P2 things. All as a placenta, I guess so. Yeah, so that's this paper here. Alright, so let me go through our next one now. Let me do one that a little more recent. Right. Actually, I feel I taken a little 5 minutes break. I'll upload a little. Yeah, I think the next one I'm going to do is a more recent one. Alright. Trying that thing here. Okay, so if you look here, so we're doing 2016. I do more recent ones. So I will run into a bunch of repeats and thing. Alright, let me know if all is and everything good. Nice. Alright, so let we go. Let me get running. Alright. Okay. Which three of the following characteristics of water of biological significance? Let's make sure it's doing good. Let's zoom out a little bit. You want to do a GC where you do a GC. The majority of people here doing keep. Right. Of biological significance. So yes, it is a universal solvent. It has a relatively high heat capacity. It's 4,200 joules per kilogram Kelvin. So no, it has a high surface tension, not low. And density decreases below 4 degrees Celsius. So this is true. Because when it solidifies, water is particular in a way that when it actually turns solid, it becomes less dense. That's why ice is float on water. So the one that we're looking at here is 1, 2, and 4. All right. So 2. Which of the following polymers is produced by the union of several of these molecules? So all remember when we was doing this, well, there's glucose, right? But what type of glucose is this? We're on question 2. What type of glucose is this? And when we link a bunch of these together, what we gain? There's beta, right? So if you look here, this is beta glucose, right? And zoom out. Oh, sorry. Nice. Right, so this is beta, and it'll be cellulose. Yeah, right. But remember how we say it's a 180-degree flip through each successive one. All right, 3. A diet high in triglycerides. Triglycerides is basically fats. May lead to obesity. The feature of triglycerides, which most likely contributes to this, is that they were well, they feel. So you have a diet high in triglycerides, and triglycerides were high in energy, right? Yes. So they have many carbon hydrogen bonds. So if we was to depict a triglyceride, all right, just now find a picture there. Okay, so if we look here, well, it has so much crap on the screen. That's not. What was this? Take this out. Take it out, COVID thing. That's not. The That's not. Just trying to pull up. Right. So if you look here, like a triglyceride, right? You have plenty of these carbon hydrogen bonds. You'll have long strings of this. And when these bonds break, that releases a massive amount of energy. And they're easy to break because there's hydrogen bonds, right? And so when we have a triglyceride molecule, and because of the number of this, and some of them would be double bonded, if you have if they saturated or unsaturated or whatever. We won't get into that now. But because of this large number of bonds, that makes them have contain a lot of energy, right? So, right. So if we look here now, look at this one. Here we have two amino acids. Which of the following bonds is formed between these molecules during a condensation reaction? Well, when we have amino acids and they form, we get a polypeptide, right? When they undergo a condensation reaction, sorry, we get a polypeptide. So what would happen here is that we would get this, right? This water being lost, right? And what we would end up getting is this carbon bonding with this nitrogen here. And that forms a peptide bond. So B, peptide. All right, so 5, collagen exhibits a secondary level of protein structure and organization. So when we have a secondary level of protein structure, it just means that we have a bunch of primary structures, basically amino acid chains or polypeptide chains linking together in a way. So the answer that we want to look for here is that not that it has 4 polypeptide chains, because each polypeptide chain is a what's call it is a primary structure. But if these chains could interact and form bonds between them, right, then we would get a secondary level. So what we're looking for here is C. Each chain forms a helix held by hydrogen bonds. And if you remember from the last thing, is a triple helix it forms. All right, so look at 6. When a mixture of starch and amylase is tested with a solution of iodine and potassium iodide, the blue-black color, right, is observed. However, when tested after 1 minute, the blue-black color is no longer observed. This is due to the formation of maltose. So starch breaks down into maltose. That's actually pretty easy. 1. Maltose, by the way, even though it's a disaccharide, it's still a reducing sugar. So if you used to do a Benedict reagent test on it, without all the acid and the neutralization, I think you would actually still get a positive reaction. Alright, so which of the following apply only to intracellular membranes? Okay, so we have two types. When we have organelles that are double membrane, like a mitochondrion, for example, yeah, they have what they call an intracellular and an extracellular membrane, right? So basically, one on the inside and one on the outside. So let's say which of the following apply only to that. So we have allow separation of cell contents from the external environment. Right, this is a kind of tough one. So let me break down each one. So yes, they do. Yeah, they do, because they would separate, because the external one would, but the internal one would also act as a barrier as well. So yeah, we could say that this one is true. Enable separate, yeah, this is true. Where you'd have little folds and things. Support metabolism, yeah, this is true, because the cristae and the mitochondria and all that. So the thing is, allow separation of cell contents from the external environment. A little torn on that one because it's true, but when they say only, I feel it's C in a. I don't feel it's one in a, because this one is. I mean, this one is not. There's both of them do this. I feel it's C. What do you feel? That's what I think. I feel 1, 2, and 3. I feel 1 semen a little could apply to the both. So yeah, I think C is the better thing. These two correct for sure. C, this is C. If all I get wrong, if it come and all I get wrong, don't blame me. All right, which of the following is not typical of a bacterium? Okay, so DNA, ribosome, cell membrane. I think we get this one. Nuclear membrane, right? So it don't have a nucleus. So yeah, the outer one would do that too. So I think logic would be C. So eight is we got this one already. Nuclear membrane. Yeah, bacteria don't have a nucleus, so you don't have a nuclear membrane. All right. We kind of no, there's not the same one. Item 9 refers to the diagram which shows a cross section of the root in a die cut. Which of the following correctly identifies structures labeled 1, 2, and 3? Okay, so all remember, say if you look at this little X structure, this is the xylem, right? So 3 is the xylem, 2 is the phloem. The phloem just be surrounding it, and 1 is the cortex. So what had that? 1 cortex, 2 phloem, 3 xylem. The docs, all they asked me if I saving docs by was saving more by. I tell them all I just jump. I just make up my mind this morning and I say, look, let me help these children and them. Yeah, so is he. Which structure is found in both a prokaryotic and a eukaryotic cell? All right, so prokaryotic cells would not have double membrane organelles. Well, so they wouldn't have a nucleus. So that knock out B, C, and D. So they would have ribosomes. Well, remember what I told earlier. The difference is the size of the ribosomes and the density of the ribosomes. Prokaryotic would have 70S and eukaryotic would have 80S. Alright, so look at 11. The table below shows 3 processes involved in the movement of substances across cell membranes. Which of the following correctly states whether the 3 processes are active or passive in nature? Active means that they expend ATP. Right? To undergo the process, passive means that they don't require any ATP for the process. So diffusion we know is passive because it doesn't use any ATP. Osmosis is also passive because again it's basically diffusion of water through a partially permeable membrane. Endocytosis takes energy. So the answer here would be C. Yeah, it would be C. So diffusion is passive, endocytosis is active because it requires energy to form them vesicles and them now. And you remember the vesicles had a move. They had to undergo bulk movement, so they had to move. The others really just occur due to the kinetic energy of the molecules. So 11 is C. Alright, 12. Which of the following is an example of endocytosis? Um, well, endo means you're taking in, right? You are taking in substances, right? You're taking something into the cell. So which one here tells of a phrase that describes something take it into the cell, take it in? A, yeah. So engulfing of a bacterium by a phagocytosis, right? So because what they're doing is that they basically giving it a big hug. They're giving the bacteria a big hug and taking it into the body and they're gonna break it down and digest it. Phagocytes are basically the policemen inside your body, and what they're doing is they're like little, like Pac-Man. Each one's like a Pac-Man inside your bloodstream, and the bacteria is like ghosts, and they're gonna eat it. All right, which of the following is the best example of anabolism? Anabolism is when you have monomers becoming polymers, right? So small, basically single units becoming multiple units. So the best example of anabolism here, by the way, anybody put it in the chat now, what's the opposite of anabolism? So this would be C. Amino acids joined by peptide bonds to form a polypeptide. Yeah. So you basically have a bunch of different single unit monomers, amino acids that's gonna get a bunch of peptide bonds, right? They can link together to form a longer polypeptide chain. So yeah, catabolism is the opposite. So catabolism would be like a peptidase or a protease that is going to break down a polypeptide back into amino acids. Okay, so 14. Which of the following graphs best shows the effect of pH on the reaction velocity of an enzyme? So the effect of pH. So this is similar to the temperature graph. And I'm gonna do a big zoom out here. Right. So think about how the temperature graph looks, the activity versus temperature graph. What which one there is? So it could be D. So if you look here, I don't know what B is supposed. I don't know what is this. I feel there's like stabilizing selection. Not stabilizing, disruptive. Anyway, so here you would have like a neutral pH, which you'd have denaturation occurring after the optimum. It gets steadily higher to reach the optimum, then goes right down. All right. 15. The decline in the rate of X is most likely because of what? Well, this denaturation, as I was just saying, and what's happening is that the enzyme. Remember, I was telling you about like non-competitive inhibitors and all kind of thing, and the enzyme itself just change shape, and the substrate can fit in there again. So which one all I think this would match the best here? The decline in the rate of X is most likely because of what? Yeah, nah, not secondary. So it could be wait now, what going on there? CXC the alphabet or what? What going on there? Yeah, okay, so is this hey, imagine like you're writing CXC, right? Like you're writing cape, right? And you're seeing as the tush, you're seeing as that, right? And you put on B. You put on B and you get it wrong, yes? Because you're not really watching the letters. You're more watching like the order, like A B C D. Way. Oh, anyway, so look at this one here. Alright, I'm gonna do some more then. Alright, so item 16 refers to the following diagrams of molecules which are involved in the formation of nucleic acids. Alright, so here we have which two molecules combine with a phosphate group form a RNA nucleotide. Okay, so you need to remember that RNA stands for DNA, right? Stands for deoxy. The D stands for deoxy, right? Which means that it lacks an oxygen. So if you look here, if you look at this group here, this here is an RNA because it has an oxy. And this one is DNA because it's deoxy, it doesn't have oxygen here. So you're looking for the one that forms an RNA. So it'll be one and uracil, one and three. So it could be A. Right? One and three. Everybody, I hope you all understand that one because the DNA again, you don't know to draw these and thing. But the DNA it lacks oxygen. That's why it's called deoxyribonucleic acid. Right? RNA is just ribonucleic acid, so it has that oxygen down there. RNA doesn't have thymine. Thymine is, uracil is, so, is more quickly formed than thymine because RNA is a real transient molecule. Like it not there for structural support, so to speak. It really just there to form a tRNA, mRNA, like things that function. I find RNA is a real dread molecule because I find the fact that it has so much different forms and has so much different jobs, they're just interesting. But yeah, so 1 and 3. Okay, 17. Item 17 refers to the following process of transcription occurring in a cell nucleus. The base sequence of the codon labeled X. Okay, where are they? We have CGC, right? CGC. So if we have CGC, right, the codon that would match with that would be GCG. So that would be what? I'm making sure I read that question correct, but yeah, so it's B. That one seemed a little too easy. Or little find this paper easier. Yeah, they didn't need to make this. There's a whole diagram them commission or whatever just to answer this stupid little question. Yeah, I was thinking they have more to it, but that's literally it. Anyway, which of the following statements about semi-conservative replication of DNA is most appropriate? Okay, so let me explain what semi-conservative replication, right? Semi-conservative replication of DNA is where it is basically to reduce copying errors. And a copying error that could happen in genetics is called a mutation, right? So what it basically does is that it doesn't build. Let me get my paint out here. God, I don't explain this one. All right. So what basically is happening here is that you would have, imagine there's a DNA, right? There's a DNA. There's a double helix. And, one of them running from 5 prime to 3 prime, and the next one running from 3 prime to 5 prime, right? And, you have your nucleotides and hydrogen bonds going to hold these together, right? Blah, 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 blah. Hydrogen bonds going to hold these together. Your thymine connected to your thymine connected to your adenine, your guanine connected to your cytosine, and so on. Okay, so what's going to happen here is that you're going to have an enzyme that's going to break all these bonds here. So this enzyme is going to go swoosh, I'm going to cut it like a bullet, right? Going to go right through. And anybody know the name of this enzyme, by the way? So what's going to happen here is that we're going to end up with one of these strands here. Nice. So that's called DNA helicase, right? So we're going to end up with one strand here and the next strand here. So this is going to be with 5 prime to 3 prime, and that's going to be with 3 prime to 5 prime. Okay? So what's going to happen now is that these bases still going to be here. So you're going to have your, I just drawing them like this, right? So these bases still going to be here. And what's going to happen now is this would be happening in like the nucleus. And in the nucleus now, we have like spare nucleotides. So if we had, I'll just point something here. If you have GCG over here, well, we know that this would be CGC. right? If we had ATT here, right, then we know that this would be TAA, blah, blah, blah. Now, what would happen now is that we would have free floating nucleotides. So we would have a G floating here, and a C floating here, and a T floating here, and so on, right? So what's going to happen is that they are going to bond to these here, these free floating nucleotides. And this T will bond with a A, this T will bond with a A, this A would bond with basically down here. Same thing here. This A would bond with a T and T and T, CGC and GCG. So all this going to happen, but it don't happen just so. What have to happen now is that you'll have the involvement of another enzyme that is literally building, right, the polymer of DNA, because DNA is a long string of nucleic acids. It's a polymer. So what's the name of the enzyme there? So we would have this we would have this but it's actually multiple of those in Cape level. You just learn that it's one, but it's actually it is actually different complexes of it, not ligase yet. So this is it would be DNA polymerase, and ligase will seal them will basically seal the fragments together. So ligase is like glue, right? So think of ligase like glue. So that's going to seal it. So polymerase is going to help to build it. And what you're going to end up getting, basically, I'm going to draw this as gray. You're going to get a new strand for each old strand, if you understand what I mean. So you're going to have a new one. There's an old strand. This is an old strand. There's an old one. And there's an old one. The ones that have in solid black, that's the old strands. And this one's in pink here. This is going to be a new strand. This is what was built from those free floating nucleotides. It's a new strand. So it don't make, well, how I can say this? It don't make it from scratch. You just have like a template to build off of. So when you have the helicase splitting that original double helix into two strands, you're going to get DNA polymerase is going to start to build. But when this build is built in an anti-parallel manner, so there's always build from 5' to 3', by the way. So you have what is called a leading strand and a lagging strand. You don't need to know about these intrinsics of that for keep. But, and then you end up with fragments. I don't know if you learned about this in school, but they call Okazaki fragments. So some of them, when they build in it, it kind of like one have to play catch up. So one of them would build real normal, but the next one had to build and go back and build and go back and build and go back. If you watch an animation of it, you'll see what I mean. And the ligase is actually what joins those fragments together. All right, so again, it's like glue, it's like stitches. So the basically what's happening here, this whole thing is called semi-conservative replication. And because what you are doing is building a new strand for each old strand. And as a result there, you are actually doubling up the amount of DNA. And this is what happens during interphase before mitosis occurs. So you'll notice that this old and this new, that they're actually identical. Now you have the off chance of a free floating nucleotide going somewhere it's not supposed to go, bonding somewhere it's not supposed to bond. It's very, very rare that it happens, but it happens. And that is usually result in a substitution mutation or some kind of frame shift. Now remember what's going to happen now is that after replication and blah, 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 what happens is that you want to make an mRNA molecule, which would then get sent to a ribosome, which would then turn that into a protein. So it basically it goes like this. You have nucleotides, then codons. Let me put it here. So you can think of the order like this. Oh shucks. You can think of the order like this. You have nucleotides, right? These form codons. These codons form genes. Those are sequences that produce a protein. Those genes, right, would then eventually go to the what happens is that transcription will occur and form an mRNA molecule. Then translation would occur in the ribosome due to tRNA. That would make a you're confused. That would make a protein, and a protein would express a phenotype. Okay. Let me break it down. Let me break it down. Imagine that you are, if you are dark skinned. Imagine, right? So whether you're dark skinned or light skinned, what is the name of the protein that gives you your skin color? What's the name of the name of the protein that gives you a skin color? Melanin, right? So we have melanin. Melanin is a protein, right? And the more melanin that you have is the darker skin that you would, you would express. So you have melanin. Melanin is a protein. All proteins are produced by DNA, by tRNA, right? So let me draw the whole story. Here we have, right? So here we have a DNA molecule, right? So we have a DNA molecule. And what's going to happen now is that here we have these bases attached to it. These are the bases. Each three of these is called a codon. So that could be it could be CAT, ATC, whatever, right? Now, this now transcription is where you build an mRNA molecule from this DNA strand. Okay, so you get something called a replication fork that's being built. Basically, kind of like how helicase split the DNA strands in half. And what would end up happening is that you would end up building this mRNA molecule. So this mRNA molecule, let me just say that, let's give you an example. If this is CAA, right, this would be GUU. Okay? So this mRNA molecule is basically like a little instruction sheet. So this is like instructions. This now would then get sent to a ribosome. So a ribosome, there's a ribosome. There's a ribosome, right? Ribosome does have basically a little ribosomal cleft here. And what's going to happen here is the mRNA going to go through this thing. Right? So the mRNA going through this thing. So each codon that's going through here. So let me say we have a codon that is GGG. Right? We have a codon that is going in here, GGG. The opposite, basically the paired codon to that, which is called an anticodon. Well, the anticodon is going to be CCC. So what is floating around out there are basically these other molecules that are called tRNA or transfer RNA. What exactly are they transferring? They are transferring an amino acid. Yeah? So they are transferring an amino acid. There's an amino acid. So they have a little amino acid attachment site. So you would have plenty different types of tRNA. Right? So you'd have one that have a alanine, you'd have one that have a leucine, you'd have one that have a cysteine. So you have different types. So let me say this particular one has this anticodon down here, CCC. So what's gonna happen here is, and I'm gonna draw it. What's gonna happen inside this ribosome? What's gonna happen inside this ribosome, right? Is you're going to have your mRNA molecule coming through there. And there's your this is what reading here. This is GGG, right? Then you would have the specific tRNA molecule, this one, that going to have CCC attached to it because this is the one that bonded, right? And what's gonna happen is that it's going to attract this one, and you're gonna have amino acid here. So imagine this now. This here going to deposit amino acid here. Then you're going to read the next tree. You're going to read the next tree, right? And the next tree, it will then have, let me say that the next tree now is, I don't know, AGG, right? So what's gonna happen is that you're gonna get a next, when this go over there, you're gonna get a next tRNA molecule that is going to have attached to it UCC. And this is now going to swoop in, and this amino acid is going to attach to this, and that will attach to our next one, 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 until we get a polypeptide chain. So when the polypeptide chain form, it's gonna fold and get bonds and wherever, and gonna form your melanin, and dependent on how much DNA you have that code for melanin, you will get many melanin molecules, you'll get a few, so, and then that would then go to your skin, your melanocyte layer, form these melanocytes that's going to tell you, well, this person dark skin, this person light skin, this person a brown skin, or whatever. So it is very, very complex. How they find it out? The smart. And what's happening is that, think of it like this. I like to think of it like this, right? All right, hold it. I need a minute. Again, a phone call. Give me a minute. Hello? Yeah, I just need a call. Yeah, woman. Nah, let me check and let me check my phone. Yeah, tell me, what's going on? You cut it? Ah, right. Okay. Okay. Yeah, 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 okay. Well, call me when you come. Yeah, yeah. Well, call now when you leaving. Yeah, 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 yeah. All right, cool. All right. Wait, so what that? So what's gonna happen here is that you're gonna have a long chain of instructions. So how I like to think of it is like, think of it like this. mRNA's instructions. I'm doing a little explaining as we go, and I'll probably don't mind. I know it's multiple choice, but still. mRNA is instructions, right? tRNA is what the ribosome is what reading the instructions. tRNA is what transferring the amino acids to the ribosome based on the instructions. And then what they're doing, that they're building a protein. Zoom out. Right. And that they're building a protein. So this here, the formation of mRNA, this is called transcription. And building the protein is called translation. Because literally what it's doing is that it's taking them instructions and making something physical out of it. It's making a protein, it's making melanin, it's making hemoglobin, it's making keratin, it's making amylase, it's making pepsin, it's making insulin, it's making whatever. Right? So that's really what's going on here. So I know I went off on a low tangent there. But we're preparing for Thursday and Friday, not just tomorrow. So what's going on? That a little easier to understand, or it's still baby on it? Transcription. All right, that's not. Let me share some notes here, right? Let me share some notes with you. All right, so if we all right, so let me look here. Let me look here. You see if Rita asks, Rita like she's get everything correct now. Hey. All right. So I don't know if you could see everything here. Make this a little smaller. Right? So the DNA code is copied onto a molecule called messenger RNA. So what you're seeing here, this is transcription. Basically forming this mRNA molecule, this complementary mRNA molecule to this DNA strand. It copying. So like if you have ACC, it can form UGG on this mRNA. And what has helped to do this is a molecule called RNA polymerase. Translation, all right, I'm going to zoom up here. Translation is where these codons now go into form proteins. So UGG will form tryptophan, GGC would form glycine, UCA would form serine, etc. And this sequence of amino acids is going to be a polypeptide, and that could eventually fold and form all kind of bonds and form hemoglobin and form keratin and form melanin and all kind of thing, right? So translation is basically taking the codons from the mRNA to the ribosome. The ribosome going to say, hey, UGG, we need a you just pair with A. So we need a ACC to go with that. So look, you see that tRNA there? He have a UC. He have a ACC. So you see for that UGG, that ACC looking real good, or they pair up there, and we're going to form amino acid called tryptophan. This UCA that here, the ribosome going to say, you know what, that AGU, that tRNA that carrying that AGU, that looking good for you there. It playing matchmaker now. So it basically playing matchmaker for each one here. So it putting together something called an anti-codon. And for you to actually see, probably have a diagram depicting it. So here we seeing what's going on. So each tRNA molecule have an amino acid attached to the top. And it's going to pair up. So this UUU, this tRNA that have UUU going to pair up with a AAA. This tRNA with a CUA going to pair up with a GAU. And so on. And each time it pair up and it go to the ribosome, it's going to deposit an amino acid. And it's going to form a little peptide bond. Why are the bases different lengths? Are they? That's probably just how the diagram is, I don't know. But it is carry a codon, it is carry an anti-codon for each codon. So, yeah, that's really what's going on. Alright, let me see if I could finish this here. Because if you just know how to go and cook. Nah, that is probably how the diagram look. The base is different lengths. You just know how to go and cook. The wife coming home and have no food. Alright, which of the following statements about semi-conservative, right, so we're coming back here, is most appropriate? So the number of DNA molecules, so it has one parental strand, so basically one old strand and one new strand. Wow. I went into a long thing to answer that way. Alright, so look at this one here. The chart you used to find are the anticodons. Yes, that's correct. That's correct. Hunter, that's correct. Those are the anticodons. I think those are the anticodons. It has different types of charts. It's usually the mRNA codons or the anticodons. It depends on what type of chart it is. Which of the following combinations correctly identifies the stages labeled 1, 2, and 3? Okay, so here we have this is mitosis, right? Here we have the chromosomes meeting in the middle. So guess what? If they meet in the middle, that's called metaphase. Meet metaphase. Here we have them just being formed. So this is prophase, right? And then they're being pulled apart because they're angry with each other. When they're angry, it just kind of move apart from each other. So this is anaphase. So this is C. Metaphase is where you meet in the middle. Prophase is where, you're meeting up at a place. And anaphase is when you're angry with each other and you're running away from each other. So metaphase, prophase is basically like nothing. You're just de-aligning, It just forming chromatids and chromosomes. And 3 is anaphase, angry anaphase. Angry is so angry you're moving away from each other. Alright, 20. Which of the following is not true of homologous chromosomes? Alright, so homologous chromosomes, that's not. My mouse going haywire. Alright, so well, not true of homologous chromosome. So they contain the same alleles. They don't necessarily have to contain the same alleles. They could contain the same number of alleles, but not the same alleles themselves. The euchromatin and heterochromatin would be the same. Alright. But they could have different versions of the genes. So like one could have a capital B for brown eyes and the next one could have a common B for blue eyes. Which of the following events in meiosis contributes directly to variation? Okay, so basically they're asking for which one here represents crossing over. So crossing over there would be D. genetic rearrangement between non-sister chromatids. I told you. Look, give me a second. My phone ringing. Are you leaving now? Yeah. You leaving now? All right. Okay. All right. Done. Nice. Okay, so a particular breed of domestic cats can have either black, white, or brown fur color. The dominant allele, capital B, of one gene gives black fur, and the recessive allele, common B, gives brown fur. Many of the white cats carry a dominant allele, capital P, of a second gene, but the second gene, what it does is that it inhibits all pigment production. So, for example, the second gene could, I will give you an example. Let me say that second gene there tells you that you can't produce any melanin. Well, you can't have black fur or brown fur if you don't produce any melanin. So this there is basically one allele that could disrupt the expression of another. So if you have an allele that would say they are producing no melanin, then having an allele for black fur or brown fur don't matter. So when we have this type of gene interaction, right, this is described as epistasis. So epistasis basically means epi means on top, epi means top, stasis means at a standstill. It basically means it brings all the other genes to a standstill. '22 is A. Epistasis, right? All right, 23. Which of the following pairs of genotypes would produce which of the following pairs of genotypes would both produce cats with white fur? Okay, so the white fur we would need, did they say it was dominant? Is recessive? Right, the recessive allele has no effect on the color of the fur. Many of the white cats carry a dominant allele P. Okay, so dominant. So we're looking for one where you would have dominant alleles where would produce cats with white fur. So, well, now when they say dominant, so it would be A if it was recessive. So it would be D. They say the non-melanin allele dominant. So because it's dominant, it could be D. If it was recessive, it would be A. Which of the following is true of restriction enzymes? All right, so restriction enzymes, it'll be A. They cut DNA at specific base sequences. They don't always produce sticky ends. It'd be good when they produce sticky ends, but they don't always produce it. All right, so that one not too bad. 25. Despite the benefit of gene therapy, many people have reservations about its use. Which of the following concerns about gene therapy may be considered a hazard? All right, well, gene therapy basically uses viruses as vehicles, right? And all viruses bad. Gene therapy will be used to clone humans. Well, I mean, yeah, but it's probably not the closest thing to a hazard. You're going into the future there. I feel the one that would work best here is C. Viruses use to deliver DNA to the nucleus and may cause diseases. I feel this one better. Because that one is an actual hazard. Attack of the clones is not a reality yet. So it could. Gene therapy could be used to clone humans, but yeah. The better answer here is C. Viruses used to deliver, right, could cause disease. It happens. Happens with vaccines too, but it's rare. Which of the following events that occur during meiosis is most likely to result in a chromosomal mutation? Okay, what do you think this one is? So a chromosomal mutation is where you could have an extra chromosome, you could have one less chromosome, you could have an inversion of DNA in the chromosome. Yeah, it could be a non-disjunction. So a non-disjunction is where um, you end up with an extra chromosome in one of the gametes, or even lacking one. So basically, if you have 47 chromosomes, you have trisomy 21, that's Down syndrome. Or it could be, you could have Klinefelter's syndrome or Turner syndrome. So basically, when you have an extra one, or you're lacking one, so you have people who have XXY, you have some people who just have an X, So but that's called a non-disjunction. Okay. In populations of Pacific salmon, most fish are either very large or very small. This is because sperm from medium-sized fish seldom fertilize the eggs. The allele frequency of medium-sized fish is therefore very low. This is most likely an example of which of the following. So if um, we have both extremes, right? We have small and we have big, but we don't have medium size, right? So what we have there is a rejection of the norm, and that is called disruptive selection. If it was the opposite and we mostly had middle ones, but not the ones on either end, that would be stabilizing. Yeah, so it'd be disruptive. All right, 28. Which of the following explains why the biological species concept often used to define a species is limited? Okay, so we have two types of species concepts. We have the biological and we have the phylogenetic. The biological one is go with like saying that if something is a species, they should be able to interbreed. They should be able to produce fertile offspring. So if you have two horses, two horses should produce a fertile horse. If you have a horse and a donkey, which are two different species, they would produce a mule, but it wouldn't be fertile. So the thing is, when using this, it doesn't apply to organisms that reproduce asexually. So, and what happens is that some species it's very difficult to it's very difficult to tell them apart because of polymorphism and thing. But we're looking for the one here that fits the best. So so the biological species concept often used to define is limited in its application. All right, so I think it'll be A. Members of different species interbreed occasionally. I don't know which ones actually, but it's the only one here that fits. D would be a rejection of the phylogenetic one. Phylogenetic one is just like, hey, this look like this, so they must be related. So it had to be A. That's the closest one. D would be not related to the biological. All right, give me a second again, yeah? Hello? Hello? Yeah. All the outside, all right, I come in. Yeah, Ole, sorry about that. Hopefully, Ole, I drop out. All right, level at 29. We got on ready, right? I was eating a little something. What schools are they from? Let me just finish eat this thing. Yeah. Ole labs are moderated. Ole got to. All moderation should have done by July. Yeah, that was the next question I wanted to ask. Anybody paper ones one yet? It had repeats. How come I didn't go for the moderation? What are you talking about? I went. I was there. Now come on, go on. If you see XC have so much foresight to set the thing, set it in units. Alright, there you go, there you go. 29. There are various. Nah, I never dock it, I do it. I didn't stay for long. I just stayed to see that everybody thing was good. Anyway, 29. There are various isolated mechanisms which can result in populations of the same species evolving into two distinct species, basically speciation. Which of the following is an example of behavioral speciation? Okay, so uh, Pinus radiata flowers in February, or Pinus radiata flowers in February, while Pinus attenuata flowers in April. So this is wrong. This is temporal isolation. Viola avensis grows on calcareous soils, whereas Viola tricolor prefers acidic soils. So this is not behavioral, this is ecological. The flower structure of certain orchids prevents the transfer of pollen um, from another species. So this is physical, this is mechanical. So that means our answer has to be D. So what it'll be, a second there. Yeah, so what we have is behavioral. So we need something that will lead to some kind of courtship behavior or something. So the funnel web spiders only males with spray. So this is a behavioral, it's a courtship behavior. So 29 will be D. So that means that spiders that don't do this, they won't mate with them. Right, 30. In sickle cell anemia, the sickling of red blood cells is a result of a mutation of which of the following? Okay, so basically it's a substitution mutation, right? That leads to sickle cell anemia. And what we're looking for is that is a single base pair that is end up getting substituted. And it encodes for a beta globin polypeptide instead of an alpha globin. So the answer here would be B. Right? So it basically forms wait, I can't remember this boy. It forms valine instead of anybody remember? I forget it. Yes, it forms valine, codes for valine instead of something else, and you end up with a hemoglobin molecule that doesn't exhibit positive cooperativity. Glutamine, yeah, that is it. Right. So basically, what happens is that it doesn't fold and right. So it doesn't fold and it doesn't each time oxygen molecule attaches to it, it folds a little bit to make the next molecule easier to attach. So it loses the ability to do that. It ends up changing the entire shape of the protein. So red blood cells on the whole gets the sickle cell. So yeah, so 30 is B. All right. So now we come into we reproduction questions. In items 31 to 32, each labeled part may be used once, more than once, or not at all. Which of the labeled structures is the female gamete and develops into the endosperm? Okay, so the female gamete is C, right? So C, and which one develops into the endosperm? Well, that would be these polar nuclei here, right? So this would be B, right? That will form your coconut water. D is the embryo sac on the whole, and A is the antipodals. Right? You don't get any zygotic, you don't get any gametic fusion or anything with those. Those basically just there to support and give nutrition and thing to the embryo sac. All right, so 33. The most important advantage of sexual reproduction is that it be allows adaptation, leads to natural selection and adaptation to change in environmental conditions. Yeah, because so it wouldn't be A. It doesn't really eliminate the risk of the deleterious mutations. It's definitely not B, because if it's asexual and be many offspring. It's not D, because it doesn't really preserve genes, desirable genes. It could get diluted or mixed up with other ones. So the only thing that would work here is C. It allows adaptation to changing environmental conditions. So basically survival of the fittest, natural selection. Alright, let me look at this one here. Which of the following types of plants use mechanisms which guarantee cross fertilization? Um, well, that would be dioecious plants, right? So remember, say dioecious means two houses. The word dioecious literally means two houses. So you have your male plants and your female plants, and so you have your male popo and your female popo, and yeah, they can't self fertilize because they have a male one and a female one. They have to cross pollinate, right? So 34 is A. All right, 35. This one shows up a good bit too. In plant tissue culture, small pieces of tissue can grow into whole new plants. In addition to inorganic ions and sucrose, which of the following essential growth requirements are needed to make this possible? Okay, so you definitely need the auxins, because auxins are basically growth hormones, right? The thing is here is what were the others? So you have vitamins and cytokinins. Okay, so it's very easy to mix up cytokinins and cytokines. There's a difference between the two. So cytokinins, again, they help with growth, they help with development. Cytokines are basically inflammatory, like chemicals that respond to inflammation. The vitamins are going to need that, of course, because vitamins can grow. So it'll be it'll actually be all one, two, and three. Yeah, it'll be all one, two, and three. And let me get a definition for lymphocytokines. Usually, this would be involved with tissue culture, asexual reproduction. Cytokinins are basically hormones that promote cell division. So yeah, you definitely want that. So yeah, and you need your vitamins too. Your Flintstones vitamins. Yes, it could be all. It one and three. No, no, no. You need the vitamins. All right. Which of the following correctly identify types of asexual reproduction? All right. So look at putting. Well, it can't be this gamete fusion, because that's sexual. So it could be binary fission, because one cell splits into two. Fragmentation, a part breaking off and developing into its own organism. And tissue culture is a very, very quick way of making lots of plantlets from little calluses. So where's that? 1, 2, and 4. So that's B. The only one that are not correct is gamete fusion. Well, I mean, again, it could look, but we know that gamete fusion wrong, right? And A, C, and D have three in it, so is B. B is the answer. Yeah, yeah. All right. 37. Which of the labeled structures produce fluids which aid in the transport of sperm? Okay, so basically you're looking for the things that produce seminal fluids and semen, and let me knock out the ones that we know is not true. So 2 is out because 2 is the erectile tissue. 4 is the seminal vesicle. 1 is the prostate gland. 5 is the testes. So I think the thing that would work is 1 and 4. Can't be 3 because 3 is where you're producing pee. Right? So 3 is the bladder, so it's 1 and 4. Yeah, 37 is 1 and 4. Alright, so 38. Which of the following is true about double fertilization in flowering plants? Okay, so double fertilization is basically you have 2 male gametes, you have your tube nucleus, and well, sorry, you have 2. Let me not go into all that. You have 2 male gametes, right? And what happens is that one of them fertilizes the egg and forms a zygote, and the next one fertilizes the endosperm, to form the endosperm. So the one here that would work is D. 2 sperm nuclei fuse with an egg, that's to form the zygote, and polar nuclei to form the endosperm. This is where you'll get your 2N zygote and your 3N, your triploid endosperm. Alright, so 38 is D. 39. Which of the following is a difference between the secondary oocyte and the ovum? Okay, so just to briefly go through this here. What you have been calling the ovum since Form 5 is not the ovum. That's the secondary oocyte. The ovum is actually right, that's what that is formed right after contact with the sperm cell and right before it turns into a zygote. It's a very, very transient stage. It there for a very, very brief time. But if all remember, when fertilization of the oocyte, secondary oocyte occurs, that enables meiosis to finish, and it ends up throwing off a polar body, and the polar body gets rid of half the chromosomes. It's like basically a chromosome dustbin, and you end up with a zygote. So the answer here would be D. The secondary oocyte is at metaphase 2, and the ovum has completed the meiotic divisions. So the ovum basically ready to turn into a zygote. The secondary oocyte is basically what we just call the egg. Right, so I going through that whole story there right now. Right, 40, the nucleus of the ovum occupies a much smaller portion of the cell than does the nucleus of a sperm. This is because what? Okay, so a sperm cell is actually the smallest, the smallest cell in the body, and the egg cell is actually the largest one. And this, the ovum, what makes it bigger is because there's only one, and it has to thrive for a good while. And remember, it has to basically undergo much more metabolic processes than a sperm cell. And in order to enable this to happen, it need energy. And for it to get energy, it need food. It need food stores, and the food stores would be like little lipid droplets, something that would be inside. So the answer here would be C. All right, which one has more polar body? The the that one is a kind of tricky question because the ovum would have a polar body, and the secondary oocyte would have a polar body. But the secondary oocyte's polar body could also undergo finish up meiosis. So a secondary oocyte could have three polar bodies, basically. So I guess the secondary oocyte could have up to like three, and the ovum would only have one attached to it. Let me get a little picture here. I believe it's a secondary oocyte. I'm gonna get a picture, and I'm gonna show you here like a little flow chart or something. CXE, them real dread to say that animal and plant reproduction is the topic says. That's the whole blasted module, you know. Alright, so no, not this I'm looking for now. Right. Okay, so if you look here, so this is the primary oocyte, right? So the primary oocyte, this is be formed before birth, and this is still a diploid, right? This is still a diploid cell. So when puberty occurs, this is where meiosis now start on this, but it get arrested. Police lock it up. So after puberty now, it free to go and do whatever again because of hormones and things being produced. So meiosis starts to continue. And the secondary oocyte now, this is the when meiosis one completes, right? We get two cells here, two haploid cells. And we're gonna get a secondary oocyte and a polar body. The polar body is basically a big waste of time. You're just there to get rid of chromosomes. So this polar body, before it degenerates, right? What could happen is that this polar body could actually finish meiosis, and this could end up forming two, right? This one could form, they could end up with an extra polar body. So this could be two. May if that makes sense, but this polar body could degenerate, right? The polar body is a little weakling. Let me get a thing there. That's not, move my thing there. I'll show you what I mean. So I have a whole set of memes in it, and I just like everything in my computer, everything is just be thrown up all over the place. Yeah, I know what is what. Alright. Nothing can open. Me can't find it now. But imagine like the oocyte is like Dwayne the Rock Johnson and the polar body is Kevin Hart. So it real small, and it just is an offshoot of it. But it's just there to get rid of the extra chromosomes. But this polar body could end up undergoing finishing meiosis, and it could end up with two here. So the secondary oocyte could end up with an extra polar body. So I don't know if that makes sense. Whereas this fertilized egg now, this would, remember, meiosis finish here. So this is your other polar body getting thrown out here. So how I could draw this boy? How long I going? I don't know. Honestly, I don't know. I might take a break just now, honestly. Alright, that's now, I looking till whenever. I might drop in and drop out, like, I'll come back and go in wherever. Alright, so if all is seen here. Yeah, I feel so. Me, I burn out. Alright. Okay, so if all is see here. Me and if it's staying, I don't. Here we have the oocyte, right? There's we secondary oocyte. There's we secondary oocyte, right? And this is the polar body. There's the polar body here. This polar body could undergo meiosis. Well, finish up meiosis, so you end up with two. This secondary oocyte now, if you end up with a sperm cell fertilizing it, is going to end up forming an ovum. Right? So it's going to form an ovum. And then that ovum going to become a zygote. It's an ovum for a very short period of time. And this ovum now, this is going to form a polar body. And this polar body could sometimes be attached to it. Right? So polar body real small. So this is going to form a polar body. But you only have one. So if you really think about it, how much daughter cells we end up with? We end up with one, two polar bodies for this secondary oocyte, three, four. Because remember meiosis, it is four polar bodies. Not for me, four polar bodies, four daughter cells. May not that make sense, but it's the secondary oocyte. There's an extra one, not the ovum. So if this polar body managed to finish meiosis, sometimes it doesn't, sometimes it does. What platform I'll be doing paper twos tomorrow? Probably the same YouTube channel. Me in the mood for experimenting. So if you just look out for this YouTube channel, you might see a live or something scheduled. I don't really know. Me making no money off of this. If I was making money, I would have come up with something proper. But it'll be on YouTube because me can't stand to learn nothing else right now. Alright, so let me finish this up now and we'll take a break. People are eat. Alright, so the hormone that stimulates spermatogenesis in the seminiferous tubules is what? Well, that's testosterone. That's the only thing that works in there. Testosterone. So testosterone is basically, that giving the sperm cells the health and strength. Alright, so 42. Which of the following events occurring during the ovarian cycle? What time you come back? What time you want to come back? Probably 7:30. Right. Which of the following events occurring during the ovarian cycle is a result of the negative feedback caused by an increase in estrogen levels? Okay, so there's a kind of tough one here. When right, so basically they're asking about this negative feedback system. So if you have an increase in estrogen, right? Negative feedback means that that will inhibit something else. So we are looking for the decreased production of something. So when we have an increase in estrogen, that is going to cause a decrease in GnRH. And that is thus going to cause a decrease in FSH and LH and all kind of thing. So this is the one that working here. Now the problem is, well, okay, so Eastern Standard Time. I don't mean now it's 6 o'clock now. It's almost 6 o'clock now. Me, where are you from? I don't know. I from Trinidad. So an hour and a half from now. Yeah, no EST is Trinidad. Me, you know, no? Hour and a half. Yeah, the so I was saying this is negative feedback, but there is a positive feedback mechanism as well, where if there's a high amount of LH, there's a higher amount of estrogen that is going to cause a spike in LH. Okay, so item 43 refers to the following events seen in the development and implantation of a human embryo. All right, so what is the correct sequence of events? Okay, so when we have development and implantation of an embryo, the correct sequence of events is going to start with the inner cell mass, right? Tiffany, maybe 2017. So the inner cell mass would grow. Then you have the alloanthus with the chorion and thing. Then the chorionic villi. Then the lacunae or the maternal blood spaces. Right. So it could be well, no. It's not A, no. I read it wrong. It's B. It's B. I'm talking stupidness. The alloanthus something would form as. So the inner cell mass would form. The chorionic villi would grow. Yeah. Sorry about that. Then then the alloanthus something would form. The yolk sac and thing. Yeah. Sorry about that. Okay, so 43 is B. Alright, there goes the last two. 44. Which of the following is an important function of the mammalian placenta? Okay, so the placenta is basically a nutritional storage. But it is also connected to the umbilical cord, so it's also a connection between the maternal and the fetal bloodstreams, like the chorion. So what we look in here is for something that acts as a system, right? So a system that would, you know, I was gonna say transport, but since it's say drugs, I don't think drugs would work. Yeah, LBA. Secretes hormones that are important during pregnancy. So when the placenta forms, um, when the blastocyst forms, it releases something called human chorionic gonadotropin, or HCG. And what that does is that it allows progesterone to keep forming. And there's placental hormones here. So part of it is that, and let me just look up an example. I think that's the one that's seemingly closest. You see, D could work too, but I don't think that's the best answer because the placenta does produce progesterone and thing, you know, it does produce estrogen and thing to help support the endometrium. So it wouldn't be C because C is the amnion. It wouldn't be B because it doesn't act as a storage point. It acts as a transfer point, but not a storage point. Um, 43, yeah. 43 is B. Now I don't think drug transportation is the function that we're looking for. So I think that it we no, the placenta does do it. The placenta does do the does release the hormones. So the placenta does actually produce those two hormones here. On top of others, it produces HCG and thing. The HCG is what, when women do a pregnancy test, exactly what's tell them, yeah, you're pregnant. It also have a next one called HPL, or human placental lactogen. Basically, what is help to produce breast milk. So it does produce a lot of hormones. I think E is the best answer to go with 44. Um, 45, this has come up a good bit too. Which of the following best explains? Yeah, they call it chorionic gonadotropin, but is okay. So all right, so there's the thing. I don't know how to pronounce the name, but there's the thing idod. The chorion is kind of part of the chorionic villi is kind of like part of the placenta, you know. I mean, because the placenta is the contains those intervillous spaces between the maternal and the fetal bloodstream. So this human chorionic gonadotropin would be counted as produced by the placenta too. So yeah. Anyway, so this last one here, which of the following best explains why women should increase their intake of folate here? So it reduces the chance of spina bifida. Spina bifida is basically like uh it's like kind of like a very very curved spine, and you need folic acid to to help to produce the neural spine. And if you don't have that, you get spina bifida. Okay, so what I'll do is I'll leave this live stream on, I suppose. And I guess I'll go put a little message here. I really don't know. Okay, let me see. What is the time in EST? All right, so it's 6 p.m. in EST Eastern time, right? I had no idea it had Jamaican something in here. So what we're going to do is just pop a little sign here. All right, that's now it. Or pop like a little text or something. All right. Hopefully current I cut off. We get a little picture here. All right. Let me see what I could add to this. All right, told you. So I'll be back in an hour and a half. Well, hello again. We'll start back in a couple minutes. Look, I try not to say anything political, but Kamala real out of touch, boy. Like, I mean, both of them, eh? Kamala was like, why are they not taking on my press releases? Or they taking on Forklift Man? Or they taking on Brownie the dog? Or they taking on Michael B. Jordan, Juve? Or they're taking on, or they're taking on what the UNC had to say. I say I was trying to keep things apolitical. Oh God, man. Well, they're taking on Brown. Where Brown you did oh, I get a picture there. Alright, you can start in a little 3 minutes. What we say we do in the 2017, right? Well, they're taking me on what I have to say, or they're taking on Brownie the dog. That's what she say. Lady actually say that, you know. Actually went by the mic and say that, oh, they're taking on Brownie the dog, but not me. Kamala. Oh, I had a caption to this, eh? Hey, it's not me who writes it, and I don't really care. It's all there. I mean, I hope it repeats. So, I mean, just in case it's not on, that's why I'm going through the little bit of theory every time we do it. But check now. It have a science. It had a science multiple choice that went yet. We ain't know yet. Assuming tomorrow is the first one. So, we gotta play. We gotta pray. They don't say we get them the P2 topics already. Hey, we gotta get them new paper one questions. Hopefully not. Yeah, if all you know anybody doing a little late night studying and things, send them this clip. We go until a little late. I mean, as much as we could do. I'm get all them thing up me, eh? Alright, that's now, eh? When you reach the P2, we go CS. Alright, that's now. Um, Yeah, man, I got the repeats. We gotta do a little praying. We gotta do a little praying. We have to do a little praying for tomorrow and make sure it have a whole set of repeats. We have to let me see who we get in the we had to sacrifice some people there. The thing, what else we need, boy? We need to perform the little ritual. What else we had to put at the altar? We had to make sure pure mats, me and about pure mats. They all the problem with that. Pure mats. What else we put? Might get in a little trouble for this one, but nah. So, yeah, we could probably get away with sacrificing Pandy because he go on this now. Go on, eh? Eh, Me I done yet. I tell you no, you walk in tomorrow and you see it and no repeats. We gotta make sure. Yeah, right, the last one, yeah. Well, if I got the last one already, what Garcia going through, Garcia going through, right? So we gotta make sure, that yeah, well, you had to pray, pray that it have repeats. You never know, eh? But I feel like I have it. I mean, if they say the business, I feel the so dread one time. So yeah, it's not us now. We had to pray that when once all they're done write this exam, that they turn around and say, we're doing a little modified marking, we're modifying the marks a little bit, because of COVID. We're going back and we're going to modify some marks. You had to pray for that too, eh? And everybody they'll be like Barbados had hurricane and St. Lucia had volcano and Jamaica have COVID and all kind of thing. And you say, it's a fear that some countries, that hurricane and thing and some didn't, so you had to go back and modify all the marks, Hey, yeah, like last year I didn't think that was possible, but nah boy. I mean all they went through it all in lower six, eh? Assuming so, probably some other repeating. But six, he going through. All right, let me start. I say let me see if we'll just do straight multiple choice this time. Um, I won't go through so much theory things, but all right, let me see if I could get this. Let me get this in there. All right, all right. So I have to make sure that. talk about forklifts and thing. I guess we people outside Trinidad probably don't know what we're talking about. Alright. Alright, let us get some things ready. But that last year was a mess and they still have fixed it, but life had to go on. Hey, the Gary didn't stay up for long enough. Alright, let me start there. That's now, eh? Use this by nice. Okay. Alright, so everything showing there, right? Okay. Alright, so let me go. Let me go. Pistia striatus, a water lettuce, is a rosette-like plant found floating in ponds and natural waterways. Which of the following properties of water assist the plants to float? Okay, so hey, remember, say if all you have any friends and thing revising for this exam tomorrow, tell them jump in if they want. Course is free 99. All right. So what y'all think this one is? So which are the following properties? Yeah. So it's basically a high amount of cohesion, which will lead to a high surface tension. So just to explain the difference between the two, cohesion is the ability for the molecules to bond to each other, and adhesion is the ability for the molecules to bond to other things. So if I take a bucket of water and I fling it against the wall, and some of the water won't just drop off just so some would stick. So yeah, so that's high cohesion. Nothing to do with heat capacity and latent heat and thing. That's really to do with like temperature regulation. All right, look at 2. Which of the following combinations correctly describes the structure of molecules 1 and 2? Okay, so if we look at 1, I know this this CXC thing. Let me put here on top here. Put him on top there. Right, okay. So the which are we right? So if you look here, we're looking to see we have 2 carbon nitrogen bonds, right? Is it 2? Yeah, we have 2. So you have 2, but if we count here, we have 1 R group, 2 R group, 3 R groups. So we have 3 amino acids joined by 2 peptide bonds, because we have 3 R groups, so it's 3 amino acids. And what we see in here? Well, the answer is C, because amino acids not joined by glycosidic bonds as sugars. So it had to be that. So here we have 2 beta glucose residues joined by a 1,4 glycosidic bond. And basically what you're seeing here, what is this, boy? This is not cellulose. Is it? No, it's not. This look like lactose or something. So 2, right? So that's C is C. Anything else? Alright. All I remember three. All I remember three. Give me the answer for three. Alright, let me look at four. Which of the following combinations of properties of glucose is as a result of its simple ring structure? Okay, so glucose has this ring structure as I show you how to draw. Yeah, so three is B, right? Remember them carbon hydrogen bonds, because you have so much of them, you basically have a long line of them. You'd have a long line of them. The you have plenty to break and release energy. Alright, so if you look at four, which are the following combinations? Okay, so we have one statement that falls here, by the way, for four. Which one is the false statement? Is it statement one, statement two, or statement three? Which statement falls for four? Yeah, statement two wrong. Glucose is a very reactive molecule. So we know that in fact, when translocation in the phloem have to occur, we can't send glucose. Glucose going in everybody else. So we have to convert it into sucrose. So anyway, it can be rapidly oxidized to produce energy. So that's true. It is an important monomer. That's true because it in, maltose, amylose, cellulose, right? So one and three. So that's B, right? So four is B. Yeah. So four is B. Two is wrong. Okay. So next one. What is the highest level? This question, as easy as it is, this question is real come. This one is real come. What is the highest level of protein structure exhibited by hemoglobin? Well, this one easier. Primary, secondary, tertiary, and your answer is quaternary. Quaternary is when you have a bunch of different tertiary structures linked together. Now, when you have a quaternary structure, you could also have, well, tertiary structures could have these too. But you could have molecules, they could have atoms that is not, or ions that is not part of the molecule, but they could still be attached to it. So, example is like the heme group, the ion that is attached to hemoglobin. It have a name for that type of group, by the way. Who put the name there for me? Alright, so item 6 refers to the following information. Students conduct the Benedict's test on 4 different clear solutions containing varying amounts of reducing sugar. The results indicating the degree of color change are shown below. Yes, it's called a prosthetic group. That's correct. Um, which of the following combinations show the correct sequence of the solutions? Alright, so this past paper is actually pretty easy so far. So we would start with blue, and then we basically go in Roy G. Biv, but in reverse. So it will be blue and then yellow. Well, actually, it will be blue, then green, then yellow, then orange, then red. So we're looking for 1, 4, 2, 3. Which one is that? That's B. All right, game as man correct. So, right, so that's 3. That's B. Sorry. So 1, 4, 2, 3. Basically, what's happening is that the copper ions are being reduced, and it lets out this, it gives this red precipitate. So it could actually be a quantitative test too, because you could actually leave it to settle and see how much precipitate settles at the bottom, and you can actually tell how much sugar is produced. Well, not produced, but how much it contains. Nice. So 6 is B. Hey, we have some repeats. Which of the following structures present in the cell would not be seen in a bacterial cell? Well, you wouldn't see. Have you bounce this up again? Right, you wouldn't find a nuclear membrane. Yeah, prosthetic groups. This is 2017. Okay. 8. Come on, now remember this. 8 is B is a tissue. The phloem is a tissue, is a group of interrelated cells. Alright. Okay. Here we bounce up a next one that show up quite a lot. Um, which of the following combinations correctly identify structures typical of a eukaryotic cell? So basically like a protist, an animal, or a plant. So eukaryotic cells have double membrane organelles, right? They also have nuclei. So the only thing, so you would have an endo, you would have a mito, you would have a nuclear, but you going to have a SMTS. So you going to have again? So it could be B. It going to have a SMTS 5G, whatever Samsung phone, right? You going to have the newer model, the ATS, right? So 9 is B. Alright, keep in mind that this one, right? This one will show up a good bit. Alright, so 9B. Here we have a next one that show up a lot, Bobby and he goldfish. So Bobby is given a tropical freshwater fish for his birthday. Experimenting with the fish, he basically murdered the fish. He put the fish in seawater and it dead, right? Which of the following statements most explains the reason for its death? Okay, so if you have a freshwater fish, that fish has a high water potential, right? Because it basically has a low amount of solute. Seawater has a lower water potential because there's more salt and impurities and so on in it, right? You know, it have pee and all kind of thing in seawater. You shouldn't drink it. So he basically put the fish in this thing. And if I had to just let me just draw a basic little diagram here for you, right? What are you doing? Alright, this now. Let's put this one on top. Alright, so basically it's kind of like this. So if we have a fish, there's we fish, right? So I'm gonna use my mouse. So here we have, is a goldfish. Right, so I know there's a fish, right? Don't judge. There's a fish. There's a goldfish, right? And inside here is a freshwater fish. So inside here you would have a high water potential inside here, right? And typically you want to keep this fish in fresh water because you don't want water to flow to the fish. And so instead here we have seawater, which has a low water potential, right? Don't be coasting on guppies, you know. Guppies, guppies is like the rock stars evolution, eh? Anyway, so this boy do this. So what's gonna happen here is that the fish is gonna lose water because it's gonna flow from a high to a low, right? So the water is gonna diffuse out of the fish. If all they go on, if all they, well, I mean, don't watch it now. But if all they look up, if all they have Netflix, look up a show called Alien Worlds, and episode 2, episode 3, I think it's both of them. It actually talk about, one of them have guppies, like they talk about the guppies in Trinidad, and they actually was in Trinidad. They went like Arima Valley or something, and real flim it up. It was real good. And they're real big way up and thing. I mean, people here they see guppy as drainfish and thing, but they're real resilient. Alright, so where we did? So 10 is A. Right, so water diffuses out of the fish. Fish get dehydrated. Alright, so 11. When a plant cell is placed in a solution with a less, remember this one, less negative just means higher. right? So with a higher water potential and its cell sap, the cell becomes turgid because water diffuses into the cell. So basically, if you put it in a higher water potential and inside the cell has a lower water potential, water is going to go in. We do that one already. All right, so 11 is A. Oh, shucks. I'm going to take off the fish. Yeah, sorry about that. Okay, so if we look at 12, can I please repeat? Okay, so I was saying that less negative water potential. Less negative just means higher, right? So when if you put the plant cell in a higher water potential, think of that as like the potential for water to move. So the ability, the chance that water will flow, that will have a net flow into right into something else. So if you put the plant cell in a solution that has basically a lot of water, well, water is going to go in to the cell, and the cell will become turgid. Turgid means like it's swollen, and yeah, because water is flowing. It's still there. Okay. Which of the following is not a rule of phospholipids in cell membranes? Okay, so what y'all think? What y'all think for 12? Well, so nah, it wouldn't be as so. Hmm. Nah, C does because the cholesterol in it does maintain fluidity, so it wouldn't be C. It wouldn't be B either because the vesicles are actually made out of the phospholipids. The the one that would work here, I'm gonna explain here. Nah, they form vesicles. They form vesicles. It could be D. So let me explain why. Um, Phospholipids don't necessarily move the material, right? So they don't actually work to move material in. The material would move because of kinetic energy, or they would move because of a channel protein, or they would move because of a carrier protein. The phospholipids actually, I mean, simple diffusion could occur through them, but that is because of the kinetic energy of the molecules. That's not because of the phospholipids. The phospholipids have very little to do with that. What the phospholipids do is that they act as a barrier for movement. So like larger molecules like glucose, which is 6 carbons, 12 hydrogens, and 6 oxygens, pretty big molecule, can't pass. Smaller things like carbon dioxide and oxygen could pass, right? Non-polar stuff. But the so it does maintain the cell and organelles within a fluid environment because it does act as an envelope in a way to keep the organelles inside because it forms part of the cell membrane. It's partially permeable. So the one that you're always thinking about is formed vesicles in the process of phagocytosis. So this is true. When you have a vesicle that is formed, right? Let me get a picture. Let me get a picture for you. When you have a vesicle that is formed, the vesicle is actually made up of phospholipids. All right, so let me show you here. Yeah, what I mean, right? So if you look here, this is a vesicle, right? And usually floating as an aqueous solution, and you would have your you'd have your good stuff in here. You'd have your good stuff, wherever you had to carry antibodies or neurotransmitters, I don't know, wherever. Right? So you have your good stuff in here, but the actual envelope of the vesicle is made up of phospholipids. So I tell you, some of these questions here, the correct answer is be so close to being correct. As really just come up to linguistics and syntax, but the real answer here, cocaine maybe. The All right, so let me get here out today. So the only one there that really do fit is D because they don't really work to move material in. They contain things that would move material in, but not the phospholipids themselves. All right. Okay, so 13, there's a repeat. Which of the following is the best example of anabolism? Um, so this would be C. Amino acids joined by peptide bonds to form a polypeptide. So we know that amino acids are monomers, and again, they join together to form a polymer. That's what anabolism does. The opposite is catabolism. I don't understand why men like deleting or retracting the messages, while you feel Gary Griffith or something coming to see this thing. Leave the thing up. Or if you people tracking this YouTube or what, to read all your comments. Alright, 14 is a repeat. Pepsin, which is found in the stomach, works best in acidic pH, so we answer is A. Right, we're looking for a low pH. Right? So this one here. Low pH. We're afraid to post about all kind of Gary Griffith and all kind of thing. Hey, let me the man done. Well, I think the only way the man go come for me is if I say his son can't play a certain sport. Then he'll come for me. Alright, so I'm gonna zoom out a little bit. Item 15. Alright, this is a tricky one. Item 15 refers to the following diagrams which represent an enzyme and a substrate molecule, labeled X and Y. In a timed enzyme catalyzed investigation to measure reaction rate, equal amounts of the molecules are mixed. With reference to the diagrams, which of the following is correct about the rate of reaction between X and Y? Okay? Um, pa guess day, pa guess day. Pa guess in the chat, what if you? D? Yeah, so D is correct. D is absolutely correct. Well, if you look at Y looking denatured to me, and the substrate X kind of fit in Y. Now, why I say is a tricky one is because um, I feel like this is a very bad question because it doesn't take into account induced fit because it doesn't have to fit exactly in the substrate doesn't fit exactly into the enzyme. The enzyme could actually accommodate smaller changes to occur. So the is a very very bad question, and I actually had to clarify it a long time ago to make sure. But the answer is D. It's yeah. But I honestly I would say a very slow reaction because it's still enough to fit. It just had to take time to induce fit, but they say it's no reaction at all. So yeah, the answer is D for sure. For sure is D. All right, so let me look at 16. Which of the following is found in both DNA and transfer RNA? Okay, so we have uracil, ribose, sugar phosphate backbone, and a double helix structure. Okay, so DNA doesn't have uracil, and DNA doesn't have ribose. DNA has deoxyribose. So that leaves sugar phosphate backbone and double helix structure. Yeah, so the answer is C. So they would have a sugar phosphate backbone because they would have that pentose group, that that phosphate group, and the nitrogenous bases. It wouldn't be a double helix structure because RNA is not a double helix. RNA is a single stranded molecule. Okay, so 17. I don't know how well y'all could see these diagrams here, but they're basically asking which of the following illustrates semi-conservative replication. And if I remember the long diatribe I went through before, semi-conservative replication is where you have one old strand and one parental strand, right? So basically, well, one old strand and one new strand, right? Yeah, so it could be A. Because what you want to look at, and I'll just zoom in here, right? Just so you all could see. What you want to look at is that there's one old one, which is indicated by the solid line, and I guess one new one, which is indicated by the dotted line. The others here don't really indicate. I have no idea what's going on in two. Three is wrong because here we in this double helix we have two old ones and two new ones. That's wrong. And here we just have two old ones, which is very wrong. So it could be this one old and one new, because when the DNA synthesis occurs, it remember the helicase breaks it apart, it gets the free nucleotides, DNA polymerase link it together, form a old, and it forms a new from the old. So remember the importance of that is to reduce copying errors. Copying errors still occur, but on a much, reduced basis. All right, so here we have a set of triplet codons, and the table is basically showing uh, the table is showing the amino acids that are being formed, and I feel I have to reduce this a little bit. All right. Let me take out the stupidness. Well, I still have this on. All right. Okay. So let me. Okay. So what we want to do is we want to write the RNA strand for this, right? So the RNA strand is going to be, if we just look at like the last ones and we look at the first, actually no. Well, if we look at the first one, TAT, right? So TAT, this could be, oh shucks, I put the wrong thing. This could be, A, this thing real thing, but that's not. Alright, so this could be AUA, right? So what's AUA? AUA, no. What's the I doing? T adenine with, wait, why doing why? AUA. I'm doing something wrong. That's not. I'm doing madness. This will be UCU. You start from behind. UCU. So UCU is serine, right? So serine, you work backwards. UCU is serine. So it's either these two. And if we look again, UUC is phenylalanine. So yeah, so it's D. Yeah. T changes, so it should be U. Yeah, I chain up myself there. Anyway, so it's D. Alright, so T changes, so it should be A. Where tyrosine there, way? Tyrosine. Right. Yeah. So it'll be UAU, right? So that's tyrosine. And GTA, Grand Theft Auto, that'll be CAU. And that will be histidine, right? So, well, we just have to get it first. Yeah. Explain. Well, what we doing, Miss Debbie Dean, is that we are writing the RNA equivalent or the RNA complementary codons for these what we see in here. So if I take TAT and transcription occurs, right? So transcription is going to make this into T would be U, A would be um Well, I feel like hold on. Like I chain up myself, boy. You no. What doing wrong is A. Is A U A. Well, I feel like I just confuffling myself, boy. Let me work backwards. Let me work backwards. Well, I feel like I look in T would be A. A would be what A would be, and A would be U. A U A. So we're going on with this question. Are you looking at something wrong? Like something wrong here? It's just me? A U A is the first codon. That's an error. Something wrong. Because if okay, let me work backwards, right? Yeah, something wrong. So if let me work backwards. If I say AGA, right? And the if when that get transcribed, that will be UCU. And when it get translated, that will be, and you look for UCU here, right? That's serine. So this last one is serine. And if we look here now, AAG. Yeah, AAG, right? This would be UUC. And that'll be phenylalanine, right? Yeah, UUC, phenylalanine. And if we go backwards again, we go backwards 3, CGA, right? This would be GCU. And where are they? G GCU's alanine. Look, something. Yeah, yeah. I think that one. This question is this question is anyway. But I think what it's supposed to be was D because it looked like well these ones I do here is matching. I don't know. Log D. All right. So if you look at 19. All right. So item 19 refers to the following diagram, which shows nuclear material in a cell, not drawn to scale. So here we have a nuclear pore. Inside here we have like stringy material. Your mother's sewing thread. And here we have, she weaving it into something nice and she knitting it and crocheting it. And eventually she form a nice, you know, a nice little sweater for you, right? So anyway, they're asking here what is what here. So one is a chromosome. Two is a histone because this wraps around the histones. Three is just DNA. It has a double helix, and four is chromatin here. So smart people here. So it's B. Item 19 is B. Um, I was telling you all here before that even though this is one chromosome, these are two chromatids because this is essentially one structure is a replicated set of DNA, right? So it's two chromatids, but it's still considered one chromosome. Is that error? Okay. Okay. The following diagram shows the structure of a tRNA molecule, which letter A, B, C, or D indicates the position on the molecule where the amino acid would be attached. The amino acid is attached on the well, this is the amino acid site here. There's the attachment site, that will be A. And we have a bunch of different loops, and anybody could tell me what these bonds, well, I just say it. These are hydrogen bonds. Jewel, I don't know why you deleted your message, though. That was, I mean, it's good to know that it was done already. Because, CXC trying to gaslight we here and feel all of we stupid. Yeah, so you see, this is the amino acid attachment site. So this is where you could find. By the way, what would be C? What would C be? Right. Which of the following best explain? Yeah, there it'll be the anticodon. So yeah, basically where the codons attach, the anticodon. Which of the following best explain why DNA replication is important in maintaining genetic stability? All right, so well, what we want to look for here, as I said, semi-conservative replication is really what helps with that, right? So DNA has to replicate accurately to allow offspring to inherit genes. So this is correct. When new cells are made, each must receive two exact copies of the instructions to function properly. Each must receive two exact copies of the instructions to function properly. I'll go back to that one. DNA must be replicated for cell division to produce. Yeah, so that's correct. Okay, so we left with B or D. When new cells are made, each must receive two exact copies of the instructions to function properly. Well, this is true to extent, except for like gametes, because they do inherit two copies. But would we say new cells are made? Well, if you come down to B or D, by I lean with D for this one here. Some of them I say that is be so close to being correct. I think that I feel is D. Or they could ask somebody, but some of them just be so close to being wrong. Like there's be like one word off from being correct or wrong. But yeah, I think that yeah, when new cells each must receive, yeah, because they replicate. Yeah, man. Yeah, SD. SD. All right. All right. So Y2. Well, remember DNA replication is occur, right? So when and that happens in interphase. So when interphase occurs during the cell cycle, you have DNA being replicated. So when mitosis has to occur, um, to form two cloned daughter cells, the chromatids have to be pulled apart and they have to be exact copies of each other. So I think since they're talking about replication on the whole, which would eventually lead to mitosis, yeah, you would need exact copies to get identical daughter cells and things like that for them to function properly. So leave with D if I hear otherwise all the time. That is correct. They have two sets of DNA. Alright, so let me type something here. Okay. Item 22 refers to the following diagram, which represents the relative amounts of DNA per nucleus during many cell divisions in an animal cell. Which of the following processes are occurring at X and Y? Okay, so if you look here we have DNA per nucleus. So we have 2 sets, 3 sets, 4 sets. And we're looking to see when the new cell has yes, they have 2 copies, but the 2 copies are still linked together. The 2 copies are the 2 chromatids. I know it could be confusing because it don't have double the chromosome number, it have double the chromatid number. So one chromosome would contain 2 chromatids. So something could have 1 chromosome or 2 chromatids. Yeah, so the answer would be C. When DNA replication occurs, you'd essentially have double the amount, at least until mitosis occur. And Y would be fertilization, because these would be when 2 haploid cells form. Yeah, it confusing. So the 2 chromatids occur as a result of DNA replication, but they still count as 1 chromosome. Alright. That's the confusing thing, because it is saying double up and just be like, oh, but that is actually replicated. That's actually 2 sets of DNA ready to get yanked apart. Okay, so item 23 refers to the following diagram showing a pair of homologous chromosomes during prophase 1 of meiosis. Okay, so what they're asking here is which of the following is a genotype of one of the recombinants? Okay, let me see here. So, all right, so if we look here, right, the genotype of one of the recombinants. Yes, the centromere. Yes, kind of count the centromeres instead. So if we look here, we could end up with A, B, C, common C. That could be our option. Our next option could be A, common B, capital C. We're looking at this spot here where they cross. So what could essentially happen is this. This here, if I trace it for you, this could end up getting this bit of DNA here. That could happen. Because where this joint is called a chiasma. But also this could happen. So we could also end up with this. I'll trace this one in blue. Alright, so this could also happen. So we could end up with 2 potential answers here. We could end up with A, B, common C, or we could end up with, well, common A, common B, capital C. Wait, only have one of these in the options here, which is this. So there's the answer A. So where this leg now, this is where this could join. They could join and exchange information here. Yep. Chiasmata. All right. Let me look at this one here. All right, so 24 and 25, I believe, are based on this information here. Fur color in mice is controlled by a... Alright, I'll go through 23 again just now. Fur color in mice is controlled by a pair of genes occupying different loci. Locus is basically like, well, locus comes from location, well, the Latin, whatever root it have for location. It basically means a place, like local. Capital W represents the presence of color. Think of it like the presence of melanin. Common W represents albino, which of course means no melanin. The allele for brown fur is capital A. The allele for black fur is common A. All right, so what would be this? So we have, well, if we have common W, capital A, small a, well, we know the capital A, small a means it means brown, but fur color is caused by the presence of melanin. So if we have these two here, right, and this basically means no color, which means no melanin, then we can't have brown, we can't have black. So the answer here is albino. Was the word for this one again? Put it in the chat. Was the word for this one? Okay. Let me look at let me go through 23 again, right? I'm gonna go through it one more time. Yes, epistasis. All right, so let me go through this one more time. All right. What they're basically asking for here, right, is they're asking for what combinations could occur due to this synapses, basically this crossing over. So if we look here, you see how this leg joining with this leg. We only have one chiasma, which is these two. So what could happen is that we could end up with this chromosomal information here. If crossing over does happen, right? That could happen. And if you look here, this one represents capital A. This dot here represents capital B allele. And this dot down here represents common C allele. So we could end up with this genotype. But the other possibility is this. If we look at this one here, getting the DNA from this leg, right? This here represents recessive A, this here represents recessive B, and this here represents dominant C. So the other possibility is this. It depends on how the crossing over happens. Recessive A, recessive B, dominant C. So this is not in the options. This is not in the options. So what the one that is in the options is that first one there. Capital A, common, I'm sorry, dominant A, dominant B, common C. That is it. Okay. What is the phenotypic ratio of the F2 from a cross between two F1 with the genotype WW capital A common A? Okay. Well, we know that the typical thing is 93 to 3 to 1. And you might be thinking, hey, well, they're not there, 93 to 3 to 1. But this is because you have epistasis happening, right? So what we're going to have now is that last 3, 1, those are going to be common, two recessive W's. So because we have two recessive W's, those not going to be black or white or gray or brown or whatever, right? Those are going to be, they're going to be albino. So the answer here would be C. I'm not feeling to go through the Punnett square for that too much time, right? So this is the answer here, 934. All right, so 26. Which of the following is considered an advantage of somatic gene therapy? All right. Hey, Rain, now come down here, boy. Anyway, so somatic gene therapy, what y'all think? So the advantage, eh? And remember, somatic means not germline, is basically so that it could go into the nucleus of currently existing cells. So example, with cystic fibrosis, and they deliver the you know the DNA through a viral vehicle or liposome. But yeah, but they use to treat genetic disorders. Um, yeah, that one is not too bad. Alright. Which of the following describes protogeny? So we bounced up protandry the other time, which is the anthers maturing before the stigmas. Um, but protogeny is where the females develop first. So this will be the stigmas mature before the anthers. So this is B. Yeah, right? So proto means first, gyne means female. Oh, fun little fact here, if you only ever hear the term androgynous, it basically means male and female combined. You know, you're watching some anime and thing, and some of them, you know, if there's a male or female, they have characteristics of both androgyny. Anyway, 28. In populations of Pacific salmon, we bounce up this one. So if we have the fish could be either very large or very small, but the medium sized one seldom fertilize the eggs. So this is disruptive. By the way, let me explain why this is very large salmon could, I mean, they could fight. They could fight each other and they could compete for the females. Very small ones, well, while the big ones fight, and the small ones could sneak by and fertilize the females. The middle sized one could neither fight and they could neither scurry past, if you know what I mean. They can sneak a little thing. So that they're not big enough to fight and they're not small enough to sneak a thing. So this is disruptive. So you'd basically find very little medium sized salmon, but you would get big ones that could fight, small ones that could be sneaky. All right, so 29. Mutations can have varying effects on an organism depending on when it occurs. The occurrence of a mutation has the greatest effect during what? Okay, well, the greatest effect that a mutation would have, right, would be during when the protein is being formed. So when they say the likelihood of it occurring is during replication, but the actual effect of the mutation, you would only really see that when you have phenotypic expression. So when you have phenotypic expression, that is where your amino acid going to come out like bleh, you know? Your amino acid going to chain together and going to form some kind of protein that don't look like anything it's supposed to look like. So the best answer here is A. During Let me read it back one more time. Mutations can have varying effects on organism depending on when it occurs. The occurrence of a mutation has the greatest effect during. No, it'll be translation. It don't necessarily have to be somatic cell formation. Because you can't form deformed gametes like due to non-disjunction and things. So that it do have to be somatic. So the best answer here is translation. So let me explain. And I understand why you would think is replication. Replication is where the errors could happen, right? That is where it could happen. Nobody really care about if it's ATT, GGG, CCC, whatever. Nobody really, that'll really be anything. That is just what the code is. Now, what happens now is that the code gets transcribed um, into mRNA. So yes, you have an incorrect code that will form during replication. And what would happen now is that when that goes to the ribosome, it will get translated into something totally different because of that. So this is where, yes, you will get your glutamine, you get, sorry, your valine being formed instead of glutamine for, for your hemoglobin that will lead to sickle cell anemia. That is when the mutation is actually expressed. So the reason saying that is because during DNA replication, it could have a bunch of non-degenerate You could have a bunch of degenerate codons that could form, but the likelihood that a mutation will actually express is during translation. So the greatest effect it would have is during translation. That is when you're going to actually see the mutation happening. That is basically what they're asking here. So if they say the occurrence of a mutation is most likely to occur, well, then is DNA replication. But it doesn't have the greatest effect at DNA replication. It has the greatest effect when they actually have that reading frame and they actually translating that nonsense into a nonsense protein that's going to have a nonsense phenotype. Let me know if that makes sense. Surely, well, I mean, yeah. So, is the way in which they word the question, because I understand the logic that you might be trying to go with. So I'm not sure what I mean. All right. So if you look at this here, right, there's there's Patau syndrome, and what happening here is basically the face forming in front of the we could say the facial characteristics. So all the facial characteristics behind the face. It's kind of like you invert the face. So this imagine this. Imagine that you have proteins that would code for your mucous membranes. You would have proteins that would code for your keratin in your eyes. You have proteins that would code for the formation of a cleft of a palate, right? So imagine now, yes, the child is born with a frame shift mutation, and but a frame shift mutation doesn't really get expressed until translation. Because yeah, you could see the code and thing, but the actual amino acids and the um the actual proteins and thing, there's actually form during translation. And when that forms, that is what leads to phenotypic expression. That's what tells you have um that tells you if you have blood type A, or whether you have detached earlobes, or whether or not you have freckles, or you have dimples, or you have a propensity for a certain disease. So you don't really know those things until translation occurs. All right, I'll take it. I'll take it off, right? So I'll give you a next example, something that a little that less something that less scary. So this is pterodactyly, right? And so pterodactyly is when you have an extra finger here, and this is basically where this might be in the DNA, but is only expressed during translation. Translation is what going to make the proteins and all kind of thing. They're going to form the collagen in these extra fingers. They're going to form the osteocytes in the bones. They're going to form the keratin in the nails, whatever, right? So when they say greatest effect during a mutation, they mean during the translation thing. And when they mean if the thing that read the occurrence of a mutation has the greatest likelihood, then the answer will be DNA replication. But the greatest effect is during translation, because that is when your mutation going to start to manifest, and you get your scary stuff and your non-scary stuff. Okay, so let me continue. Which type of isolation plays the most? Well, we do this one already. So there's A, right? Which type plays the most significant role in allopatric speciation? And allopatric just means it in a different land, right? So it's geographical. All right, 31 we did this already, right? As well, one, two, three. Right, auxins and cytokinins are both hormones needed for growth and cell division. Vitamins are needed for growth. So 31 is D. 32, sexual reproduction would be most advanced. We do this one too. Actually, no, we don't have the same answers. Would be most advantageous for the survival of a species if, what do you all think? Which one would sexual reproduction be the most advantageous? So remember, sexual reproduction is what increases vigor and fitness, natural selection. So is if you have a new disease, yeah, disease. If you have COVID Delta variant, enter Trinidad. Well, everybody had different immune systems, and if we was all the same, then all of we dead. But all of we is not the same. Some people get COVID and they could still go to work every day, and they're still good, and it'd be like they never get, even though they're like 70 years old and not there are no vaccine and they have comorbidities. You know what I mean? And they're still real good. Apparently, like they're still working hard. They're still doing all the ministerial duties, even though they have COVID. Some people just real good. Some of we, some of we are raindrop fall on me and we get sick. Yeah, 31 is a repeat reader. So yeah, it most advanced because natural selection. Nah, I get no second dose yet. A horticulturalist has bred a hibiscus variety which bears. Explain 31. Okay, I'll go back to just now. Has bred a hibiscus variety which bears violet flowers. Which method of propagation would be the most appropriate for rapid production of a large number of clonal plants? So, is testiculture? So, testiculture, you dig out the little calyces and that will grow into new plantlets. And it's very important to keep it sterile because they're very, very volatile, they're very small. All right, I'm gonna type up a little thing for one. All right, so auxins and cytokinins. I'm not too sure which version of vitamin B, but I believe it's vitamin B. Well, all I remember 34. 34 is a repeat. We go bounce up our repeats and as we go in, so it could be faster and faster. We're doing it. So 34 is D, and this is because this is correct, but this is wrong because these two are sexual, not asexual, right? So which of the following methods of asexual reproduction is incorrect? Either by its definition or its examples. Everything else is fine, but the hibiscus and flamboyant don't reproduce via spore formation. Okay, so 35. Which of the label structures will give rise to two male gametes? Put it down there. Put 36 one time. Which of the following does not promote cross fertilization? 35D, yeah. Where's called this nucleus, by the way? Where's called this nucleus? 35. The thing have a name. Yeah. So there's the generative nucleus, and one where's called our one. It's called the generative, and it two male gametes are produced because this leads to double fertilization, right? And this one here is the pollen tube nucleus. So that one, two is the entine and three is the exine. The where we they? The tube nucleus is what grows the pollen tube. It basically performs a process called chemotaxis. It like releases digestive chemicals and enzymes that would basically eat its way through the style of the flower, and the generative nucleus would go down, and the tube nucleus is just like, well, follow me, let me go, let me go. We go into the good place. We're going to see the egg. We're going to see the polar nuclei. We're going to make some zygotes. We're going to make some endosperm. That's what the tube nucleus telling the generative nucleus. That's what it's telling them male gametes. We're going to do some double fertilization. All right, so 36, all this is 36. 36 is A. Which of the following does not promote cross pollination, right? So our answer is cleistogamy. Cleistogamous plants are those that is the shy flowers of the plant kingdom. Here we have flowers that close up the self. They're like, nah, no, Mina, I good with myself. They are the introverts of the plant kingdom. So, example is tulips. A tulip is a cleistogamous plant. Um, I remember when they close up, that limits how much pollen is going to get out there. It limits how much insect vectors are going to be able to contact it. The opposite of cleistogamy is chasmogamy, which are basically open flowers. Okay. All I remember 37. 37 in plants, the ovary is best described as a swollen base of a single carpel. So it's not a lot of people put B as the answer, basic unit, but the basic unit of a gynosium is a carpel. But the base of the carpel is the ovary. Alright, the following are advantages. I repeat too, the following are advantages of two types of pollination. Which of the following are specific to cross pollination? Well, again, it increases big and widens the G pool, natural selection, 2 and 3. So that I see. Alright, let me look at 39 and 40. 39, at which of the regions label 1, 2, 3, and 4 does fertilization take place? Okay, so fertilization takes place in the oviduct. So this here would release the secondary oocyte, the ovary release the secondary oocyte, and you know, sperm cells would come, and that would happen somewhere in here. So it could be B. Alright, 40. How does a secondary oocyte differ from the ovum? Okay, so this is a kind of tricky one, and I was trying to explain this the last time. I forget who was asking. I don't know if it was Rita who was asking the question here. So the answer here is, let me explain it one more time. Yep. So it's C, and let me go through it again. So here we have a secondary oocyte, right? And this is going to form a polar body, right? So you're going to form a polar body. And after fertilization occurs, right? There's a sperm. Right? After fertilization occurs, what's going to happen is we're going to get the formation of an ovum. Right? So you're going to get the formation of an ovum. And this ovum is going to, again, have another polar body. But what is going to happen is this polar body, this first one that would have come out from the oocyte, right? This could actually finish meiosis. So you end up with two polar bodies, right? And I was saying, if you count it up, if you count up how much daughter cells are formed by the end of the day, we have 1, 2, 3 polar bodies and one ovum that would then turn into a zygote. Right? So the ovum would then become a zygote when, DNA join up and thing. But I say the ovum is a very short stage. So the answer here would be C. Females complicated, eh? Like that whole system just complicated, eh? Like the male one is like, hey, spermatogonia, hey, let me form some spermatocytes, hey, let me form some spermatids, hey, let me form some spermatozoa, hey, let me hook up to some Sertoli cells, and you know what I mean? Let me develop, and this the female one, the thing had to start, it had to stop, it had to start, it had to stop, it had to finish, it had to wait for the sperm cell before it continue, all kind of thing. Alright. Alright, so item 41 refers to the following graph, which shows the relative concentrations of the pituitary hormones W and X and the ovarian hormones Y and Z. Okay, so W and X, when I say pituitary hormones, they're talking about FSH and LH, right? So um, the W is FSH and X is LH, and I'm gonna tell you how you would know that. Or you see this little spike here? This is known as the L8 spike or the L8 surge. And the L8 surge is what happens as a result of positive feedback when estrogen levels are high. So when estrogen levels climb up to a certain point, this triggers a surge in luteinizing hormone. This here is actually what triggers ovulation. Anyway, actually that's all you need to know is B. But the Y and Z, Y is estrogen and Z is progesterone. So if we look at Z, by the way, the progesterone levels here, but if a woman is pregnant, right, this will remain high. But what is helped out at that point is the placenta, the blastocysts that produce an HCG and thing. But the placenta is what going to start to produce progesterone that going to keep this high, because I mean, if the woman pregnant, right, and the progesterone level fall, well, the baby going to fall out as she one time, because it wouldn't have any soil to keep the seed planted, if you know what I mean. So this here had to remain high, and it's usually placenta that keeps it high. 40 is C. C for chiasmata. Alright, 42. A sample of a woman's urine is tested at a lab and human chorionic gonadotropin, as HCG, is detected. This indicates that what? So the answer for 42 for me. And 43, which of the following pairs of structures contributes directly to development of the placenta? Right? Answer for 42 and 43 for me. But as I say in HCG is when a blastocyst, the blastocyst is the first thing to start secreting is HCG. A blastocyst would not increase, I'm sorry, a blastocyst would not form unless fertilization occurred. So this would be D, so that's correct. And which of the following contributes directly to this? So this is the chorion and the endometrium. The chorion again is what connects the maternal and the fetal bloodstreams. The endometrium is where you would find the intervillous spaces. I don't know what they call it at all in school. It have plenty names for it. Intervillous spaces. It have lacunae. I don't know, right? But that's where you find the stuff. Okay, so 44, oral birth control pills work most directly by doing what? Well, you feel for 44. 44 is B, yeah. So it suppresses basically ovarian release of ovarian things. So this last one here is a close one. Thickening of the cervical mucus is more due to like IUDs and thing. Um, but body birth control pills is hormonal, so B. All right, last one. Well, oh god, pregnant women are advised to increase their intake for folate. Which of the following best explains this advice? The fetus is at risk of developing spina bifida. Right? Same thing. So yeah, the neural spine hasn't formed properly, and that's what leads to that. And that's that one. But not bad. That one wasn't bad. 30 out of 45. That too hot. All right, we gonna do one more because all you had to sleep. If all this is a EM exam. What's the answer for 4? 4 is B, Emily. All right, let me see what I have. 2020 is probably not a good idea, because 2020, I mean, they won't bring the thing back to back. Anyway, you never know, eh? Let me see if I have 2020. Um, Honestly, 2019 is probably a better one, but let me check. Okay, so I have the 2020. All right, 2019 or 2020? Hey, I only doing one in a, and plus all you had to sleep. I can send all you the rest though. 19? No, I can send all you the 20 after. I'm not doing no 2000, no 2011. All right, I'm doing the 2019. I can send all you the 2020 stuff. All right, so I'm gonna give all him a PayPal, right? So I'll go PayPal me like a little Bobby or something. PayPal me a little coconut catch. Or they know the scene. You see these people who asking for 2009 and 2011 and all kind of thing. I was all they talking about when the thing now start, you know. All they talking about it when the thing now start. When the thing now start and they know nothing and they don't know what to put in the syllabus. Or they talk about 2009 and 2010 and thing. Or they talk about when Cape was a little baby and have on diapers. And Wayne probably like had a breastfeed it or something. That's all they're talking about. We are doing them thing, eh? It don't matter. I go link it here in the chat. Like before I go, I go link a Google Drive of the stuff. 2001. You think the name exists in 2001? Name right CXC yet. 2001 when World Trade Center them fall down. All right. Okay. Let me go there. Wayne could take it, man. Wayne real good. He real good. The man. Hey, if only watch any press conference with that man is a master, you know. That man is a master of manipulation. If I had to like get any if I have a company and the company falling down and I had to get somebody to go in front of the camera and say everything real good, everything we doing everything correct, it best I hire Wayne. That man is a master. That man could just say the live going through. He could say you could show him the thing in front of his face and he could say that is not it. That man is the best man to have for a company. And basically, CXC commit massive fraud last year. Last massive. Let's talk about work, but get out of here now. If you want to work, go and do work for yourself. All on my thing here. Hey, what tell all the free thing not good and good thing not free. Get the hell out of here. If you can't take it. Alright. Let me go. Let me start. Alright, which of the following molecules best way I just do hey I doing a little we'll talk here. All they frightened for tomorrow. Way like all they frightened so let me do the Alright. Which of the following molecules best represents the structure of a triglyceride? Okay. Well a triglyceride you're gonna need a glycerol molecule and you're gonna need some fatty acid chains, right? So which one they are looking like a triglyceride? Well, go on tell me to do work. I is not my teacher. Alright, so yeah, so it's C. Mia is not my teacher. I just doing this because I feeling sorry for all you. Alright. Okay, so why not D though? So D actually pretty close to it because D has two fatty acid tails. D is a phospholipid because we have a phosphate group here, so it's not D, right? So the answer is these R here represent the side chains. Alright, so it's B. Okay, so let me zoom up on this a little bit. During hydrolysis of the protein structure shown above, which of the following bonding interactions is broken? Okay, well this one easy too because Here we have peptide bonds. We have a polypeptide chain. We have proteins. What else it could be? All right, that's not it. The renal come down here, boy. Say I bust it from here, yes? All right, let me go again. Um, 3. All right, bounce up the same thing. Right, so CH bonds. This one here will show up quite a bit. Which of the following structures is not likely to be seen on electron micrograph in a plant cell? What do you feel is that one? What do you feel is 4? 4 is A here. Plant cells don't have a centriole. They just have a mTOC, a microtubule organization center where there's pack hampers and all kind of thing. Right? So they don't have centrioles. They just have microtubules. All right. So 5 and 6. Hey, I feel like all the papers we had number 6 in it. All right. So five is quaternary. I feel like every paper we had 8 too. Wait, look Bobby reach again. Anyway, so which structure is found in both a prokaryotic cell and a eukaryotic cell? That is a ribosome. Difference is the size. Collagen exhibits a secondary level, we bounced this up the first time, a secondary level of protein structure and organization. And this is because when you have these polypeptide chains, they're held together by hydrogen bonds. So remember, it's a triple helix, is basically 3 repeating amino acids over and over again. Forms a fibrous protein, is insoluble, used for mechanical support. Alright, the area labeled X is a tissue. And here we are the murderer, Bobby again. Yes, Missy. Wait. That's not. Let me explain something, right? So they say which one here shows a secondary level. I know you might be thinking D is made up of repeated units of 3 different, but this would be a primary level of a protein structure. Anyway, so um, Bobby Brown here murder a goldfish, and it's because water diffused into the fish. Um, wait, no, not into the fish. They flip the thing out of the fish because he put the thing in seawater, and there's a freshwater fish, so that means it has a high water potential right inside the fish and a lower one outside. Who tell me do 2019, boy? 2019 full of repeats. Alright, the following table shows 3 processes involved in the movement of substances across cell membranes. Which of the following rows correctly classifies each process as active or passive in nature? Remember, passive is where it doesn't use ATP, active is when it does. So diffusion is passive, endocytosis is active, osmosis is passive. 10 is C. Yeah, 9 is B. 9 is B. Okay, we have one here that is bounced up a little bit time to time. Um, 4 cylinders of equal mass are cut from a single potato. One cylinder is placed in distilled water and the others in zero point 0.2 moles, 0.4 moles, and zero point 0.8. Each cylinder is weighed at half-hour intervals for 6 hours. The graph below shows the results obtained. Which curve most likely represents zero point 0.4 moles sucrose solution? Okay, well, we know that the one with the 0, that's the one that's going to have a positive change in mass, right? So this going to be the 0 moles. And this one here is going to be the zero point 0.2. And as you go down the road, it's going to lose mass at a point, right? So 0.4. And this one here going to be 0.8. That's the one where it will lose the most mass. The most of the water would flow out of it. Just making sure. So it's BC. All right. Which of the following describes cholesterol in a cell membrane? Cholesterol is there as like to pack. Imagine it's kind of like to prevent the phospholipids from ganging up together, but it also helps to bring them together if they're drifting too far apart. So this question actually shows up quite a lot. So the answer is D. It enhances the flexibility and mechanical stability of the membrane. 12 is a repeat. 13 is a repeat, sorry. Anabolism, amino acids join to form a polypeptide. Alright. Way boy. Alright. Pepsin is A. There's one. Stomach. Stomach. Alright. Stomach is acidic, so pepsin found there. Oh, they're real good boy telling me to do this paper. The whole paper is repeats by. Alright. Decline in the rate of X. Right. It loses its tertiary structure. Okay. 15 is C. It loses its tertiary structure and it denatures, it warps basically. Substrate can't fit in it again. 16 is repeat. Sugar phosphate backbone is the found in DNA and both RNA. We did this one, and I was saying, yeah, and I was saying the difference is we're looking for RNA, so RNA would have the oxygen, right? Because deoxyribonucleic acid wouldn't have this oxygen here. So DNA is RNA, and uracil is RNA nucleotide. So maybe one and three. Okay. Item 18 refers to the following table, which shows the codes and the matching amino acids. Which of the following amino acid sequences would be produced from information coded on the DNA strand shown below? ATG. All right. So remember, this would be um, UAC. I feel the fixity, tyrosine, um, GCC could be CGG. So that's arginine. And 17, okay, no problem. And AAT, well, this could be UUA. Oh, it's GGC. Sorry, GG. So this would be CCG. Yeah, sorry about that. UUA, and that's leucine. So B. Well, at least this one had it correct. 17. All right. this is how with 17, right? Well, first of all, we know tree is one, right? Because uracil is found in RNA. Uracil is not found in DNA. Thymine is found in DNA. But you have to remember, DNA stands for deoxyribonucleic acid. Deoxy basically means it lacks an oxygen that the RNA has. RNA just stands for ribonucleic acid. So we are looking at this group here, right? This carbon, well, there's a carbon here, and this has a OH group here, and this has a H. So this one doesn't, it lacks oxygen. So this is DNA because it's deoxy, it has been deoxified or whatever, right? So, well, deoxygenated. And so this is why this is DNA. So this is why our answer is 1 and 3. Because RNA would have this oxygen here. It's not deoxygenated. And 3 here would be, well, uracil. So that's explained. So 19, well, oh God, man. Something wrong. 4 is a chromosome. Oh, no, no, not wrong. Let's see. Right. 1 is chromatin, 2 is DNA, 3 is a histone protein, and 4 is a chromosome. We just bounced up. Alice? You know, they thief this from the internet. I could see where they rub off the other label here. Anyway, hopefully they pay the people. Alright, 20 is thankfully not a repeat. Item 20 refers to the following process of translation occurring at a ribosome in a cell that synthesizes enzymes. Okay, so here we have the growing protein. Here we have the amino acid chain, right? The polypeptide chain, I should say. And this is the transfer RNA. There's the tRNA here, right? And this is, so was this here? The structure P is amino acid. Baby thing, baby thing. Where the girl who tell me to shut up? I say when old talk. She a typing. She just taking it in. Alright. Which of the following statements is not true of most homologous chromosomes? We get this one already. They contain the same alleles, right? They don't have to contain the same alleles. Because, as I said, some could have dominant and some could have recessive. Alright, 22. Well, here we'd have two sets of chromosomes, but during DNA replication, we'd have 4 sets. Still the same chromosome number though, but they'll have double the amount of chromatids. So when gametes are produced, so this here basically is when you have gametes. And when they fuse, this is fertilization, and this is going to form a diploid zygote. So repeat again, 22C. 23 is a repeat. If you have a dominant allele that gives black fur and a recessive that gives brown fur, and you have a recessive allele, you have an allele, sorry, that basically prevents pigment production on the whole, that is called epistasis. And as I was saying, what the word epistasis means, epi means top, and stasis means come into a standstill. So what epistasis means is, hold on, let me get a little thing there. The old talk is what make a good man. That's now, eh? Look at it. Right. So basically, what's happening here? Right. So we have phenotypic expression of, you know, alleles or something, and epistasis was like, hold it right there, hold it right there, right? That happening, right? So that's epistasis is going to stop it from happening. That could be, I mean, it usually is a neutral thing, but it could be bad, it could be good, but that's what it is. It basically it would override the phenotypic expression of something else. As I said, it brings it to a standstill. Okay, so there we go. 24, from the position and shape of the chromosomes, what stage of mitosis has been reached? Okay, what if it is 24? There's a unique one. Alright, so the next so it's anaphase, right? And just to explain it here, um, as like to give this story here. Well, in Form 5 last year, or they didn't have a grad, or they didn't have like a prom or a graduation party. But if you had one, you would basically dress up like how chromatin is dressed up into chromosomes. Prophase. Prophase is the dressing up scene, right? You're dressing up and you're just there. So after prophase, you have metaphase. This is where you're actually meeting up. Metaphase, you're meeting up in the middle. And then you have anaphase. Well, anaphase, you meet up and now you vex now. Something happened. Now, the grads don't go so good sometimes. Or they probably dodge a little bullet there. But you're vex. So the anaphase is when you're angry and you get pulled apart. You be like, no me and that. Right? So angry anaphase. And telophase is, well, eventually you're gonna form two cells. So when you're forming two cells, you're basically in two different places now. So you have to call each other on the telephone now. Right? I don't know. I just come up with all kind of convoluted ways to remember things. But I just remember like that. You dress up to go to prom, then you're meeting up, then you're angry, and then you have to call them on the telephone, Yeah. So yeah, prophase, metaphase, anaphase, telophase. But this is anaphase because they're clearly angry with each other and moving away from each other. It's not telophase yet because they don't have a nuclear membrane forming yet around these chromatids. Yeah, anaphase, you carry a date and you date a dancer into the dance with everybody else, but you must get vexed. That's anaphase. Anyway, 25. Mutations may arise due to non-disjunction of chromosomes. So remember saying that is where you end up with an uneven number of chromosomes in one cell. So you could end up with 24. You can end up with 24 in a cell and 23 in the next one, for example. In which of the following processes would such a mutation be transmitted to the offspring? Okay. Well, so for 25 here, where would you usually see this? Well, it would be during the formation of a gamete. So it would either be the formation of a sperm cell or egg cell. So the only one here that really fit is D. During the development of the secondary oocyte in an animal. Yeah, it would be during gamete formation. A possible benefit of producing transgenic plants is improved human nutrition, like with golden rice. Right, so 26B, or you have plants that is produced doing pesticides. Alright, 27 is non-disjunction. Get this one already. Which of the following events that occur during meiosis is most likely to result in a chromosomal mutation? Non-disjunction. Skip love the salmon, boy. Salmon, this is good, eh? Anyway, so it's disruptive selection. Feel like, eat a little salmon sushi. Hey, I real miss sushi, boy. Kit Raleigh open back the country now, but at least open the sushi and them now, boy. Open Kaizen. Aim for a little delivery. Anyway, so it's disruptive. Right? You have big ones that could fight the other big ones. You have small ones that sneaking past middle ones. Hard lovely. Alright. Item 29 refers to the following map of a plasmid showing the position of recognition sequences of two restriction enzymes. In addition to the human insulin gene, which of the following enzymes would be required to produce a recombinant plasmid? Okay. Well, if you remember, restriction enzymes cut the thing. Remember, say restriction enzymes is like genetic scissors. What here would be like genetic glue? Yeah, it could be like DNA ligase is what would actually hook them sticky ends and things together, make sure that the plasmids get taken up. Well, the plasmid, it don't make sure the plasmids get taken up, but it's make sure that it's sealed. When it cut the thing with the scissors, it glued back together. Borders close until the 14th. I talk about no borders, I talk about sushi. I don't really care about KFC and thing, but I really missing some nice food boy. KFC and think stay right there. Man, never buying KFC again. KFC them give the people a little aloo pie and a Coca-Cola and they say all they live on that for a month. I can support them thing. I go fry my own chicken. Yes, they combine the plasmid DNA and the new DNA to make recombinant DNA. Alright, 30. Which type of isolation plays the most significant role in allopatric speciation? Same thing, geographical. I know vex, I had to cook every day, I know vex for that, I like to cook. But it has some things I cannot make for myself. Right? I don't care about doubles and thing. I don't care about them thing. But it has some things I can't make it. I try and I can't do it. And I'm missing it. Which of the following structures are formed during development of pollen grains? Okay, pollen grains. What do you think? There's a new one. Now, I mean, we just went through it. Remember, the pollen grains have two nuclei in it. They have a generative nucleus and they have tube nucleus. Right, so the answer is tube nuclei. The antipodals, the synergids, and the polar nuclei are all found in the embryo sac, not the pollen grains. And surprise, surprise, here we have embryo sac. Which is alright, so which of the following structures, labeled structures develops into the endosperm? That is two. The polar nuclei. One is the antipodals, three is the egg cell, four is the, I don't know, assuming a vacuole. Well, tell me. Alright, 32B. 33. Which of the following terms is most appropriate to describe a plant in which the anthers of the flower mature before the stigma protandry? Repeat. Which of the following statements is true about double fertilization in flowering plants? We did this one too. Two sperm nuclei fuse with an egg cell and a polar nuclei, respectively. Yeah, so I feel this one go bust up just now. I'll be like, do 2019. Which of the label structures will give rise to two male gametes? That is the generative nucleus. Just do that. That's the. Hey, this is the same paper. Like, we bounced all of this up already. Which of the following conditions does not promote self pollination? Cleistogamy. In plants, the ovary is best described as the swollen base of a single carpel. Which of the following statements identifies the difference between the secondary? Now do this. Oh, it actually no wait. They just shuffle it. It's like the same thing. The secondary oocyte is at metaphase two, while the ovum has completed the meiotic divisions. Yeah, you know, B B could be correct too. No. Anyway, 38 is D. 39 oviduct. Fertilization occurs in the oviduct. 40 human spermatozoa have to spend several hours in the female genital tract before they can fertilize an oocyte. During this time, the spermatozoa are activated in a process called capacitation. This is basically they become supercharged and they become real determined to fertilize the egg. But that's basically because they lose a lot of mass. So the acrosomes, which is like the enzymes in the head, they start to activate and they're ready to basically eat through the egg. That's called capacitation. All right, so we just bounced this one up. Which of the following events occurring during the ovarian cycle is a result of the negative feedback caused by an increase in estrogen levels? So that's mm, all right, so that's D. Yeah, D. Decreased secretion of FSH is a negative feedback, so I increased later, I decreased. We just did that. We just did this one too. A sample of a woman's urine, test for HCG. This indicates that the woman is pregnant. So implantation of an embryo has occurred. And 43 and 44. Which hormone stimulates the development of the endometrium? And which hormone regulates the precise time of ovulation? Paul answers there. What do you think? Okay, so this one is A. I'll explain why this one is not progesterone, by the way, right? Which hormone regulates the precise timing? This is during this L8 spike, right? So that's C. So progesterone doesn't help with the development of the endometrium. Progesterone helps with maintenance of the endometrium. Estrogen is what helps with like follicular development, is what helps with the development of the endometrium itself. But progesterone, which is secreted by the corpus luteum, which is secreted by the placenta, is what will maintain it, is what will keep it there. So A is the better answer. It keeps it tick, that makes it tick, right? Okay, and here we have this last one. Item 45 shows a transverse section to the middle piece of a sperm cell. The structured label H is what? Well, that's basically like stuff that will connect to the flagellum. And because the flagellum will come through, the middle and taper down to form that thing that will give it motility. So This is a microtubule. The actual name for like this whole section is an axoneme, but they basically a bunch of microtubules. Yeah, man. So that it. Then I guess we could do one more. 2020 is a set of repeats too, you know. Check out all this. Check out all this. We don't know do all of this. We know do all of this. I go link all you up for the 2020 as we done. What about 2018? This copy of 2018 I have is real mess. Look, it's the same thing. It's the same thing. Alright, let me. I feel we should do it earlier, you know. What if we pull out 2015? No, we do 2015. Did we? I just know every paper I look into, I see in Bobby and the goldfish. Alright, here what? I feel it's not a wise idea to do 2020 because how it was last year now. Let me do the 2018. Let me do the 18. Let me do the 18. Let me do the 18. Honestly, it don't matter which one we do. We're gonna bounce up a whole set of repeats. And you know what I'm gonna do? I'm gonna skip the repeats. I gotta go through them like real fast. Alright, that's now, eh? Alright, let me go through it quick. This is the last one. Which of the following statements about smoking indicates that it does not contribute to CVDs? Alright, so I have no idea why this question is here in Unit 1, by the way. So, but the answer is B. Carbon monoxide decreases the heartbeat. That is not necessarily true. It reduces how much oxygen is taken up into the bloodstream, but it don't necessarily reduce the heartbeat. All the rest here basically could lead to CVDs, right? Increasing blood pressure, clogging our arteries, damaging our blood vessels. Yeah. Right, carbohydrates are transported in plants mainly in the form of sucrose. We just do that. Alright, 3. Here we have same thing, alpha and beta glucose. Right, so 3 is D. We just bounce this up. Structure 1 contributes to starch as alpha. Structure 2 contributes to cellulose as beta. Alright, so 3 is D. Alright, 4. Which of the following arrangements is the correct orientation of heads and tails of phospholipids in a bilayer which facilitates membrane stability? Well, tails are hydrophobic and heads are hydrophilic. So, what y'all think 4 would be? Right? Tails are hydrophobic and heads are hydrophilic. Right? So it'd be D. Heads would be attracted to water. Tails would be repelled. Will go away from water. At 5, it just bounces up collagen secondary levels because it forms a helix held by hydrogen bonds. How much people it have in here? I feel everybody went to sleep. Everybody in here again, I want it. Everybody who went to sleep, them again at 3. The chemical structure above represents a molecule of what? This is a tripeptide. Again, you're looking at CN, right? These are peptide bonds. So there's a tripeptide. Which of the following is not typical of a bacterium nuclear membrane? Wait, boy, I feel like the 5 papers we do this question come up. All right. People getting tired. Which of the following statements best explains the function of the region label 1? Okay. Well, oh shucks, I forget 6. When a mixture of starch and amylase, there's a repeat too. This is due to the formation of maltose. Starch um, hydrolyzes into maltose. Hey, I don't know why you're looking at here now. That look like something. Uh, is a nucleus? Is a nucleus. So there's a nucleus. There's a nucleolus. There's a nuclear pore. Um, which of the following statements best explains the function of the region labeled I? So that's a nuclear pore, right? Well, the nuclear pore is basically like, think of it like a channel for nuclear material to mainly exit the nucleus. So, like, if you have a mRNA molecule, let me say that there's a mRNA molecule. This mRNA molecule could ship out through here and it could go into the cytoplasm and it could find a little ribosome, you know what I mean? Find a little ribosome, make a little translate and make a little polypeptide, you know what I mean? Make a little protein, make a little phenotypic expression. So it's D, right? Right, so it allows for the transport of mRNA and ribosomal components into it. So wherever in here does the doorway it could like exit. All right. 10. Which of the following combinations forms evidence which best supports the endosymbiont theory? Okay, so the endosymbiont theory and the endosymbiotic theory basically tells you that eukaryotic cells originate, well, life basically originated with um, having having early forms of chloroplast and mitochondria um, be integrated together to form a super molecule, you know what I mean? Well, not super, but a normal molecule, and to form a normal type of cell. So in prokaryotic cells, you would have circular DNA in the cells, right? It has basically plasmid DNA. And eukaryotic cells, circular plasmid is actually found in the mitochondria. So you're looking into the one that match. So because they have the same type of DNA in the two, right? Because they are plasmids in the two, that there is what best supports it. So it be B. But what really the theory tells you, endo means inside, and symbiont comes from symbiotic, which comes from symbiosis, which basically comes from things working together. So it basically saying that an early cytoplasm would have absorbed an early chloroplast and be able to photosynthesize early mitochondria and be able to respire. And that there would have formed the precursor for the cells that we know today. And they say that there's some leftover evidence that is still there. The fact that mitochondria have their own versions of ribosomes. The fact that they double membrane, which will insinuate that there was once their own thing. So yeah, so 10 is B. 11. In which region would vascular tissue associated with the transport of material be located? Okay, so here we are looking at a root. A die-cut root is not a very good diagram, but we're looking at a die-cut root, and let me zoom up here a little bit, right? Just so you could see. And this here, if I could trace it, this is the xylem, and the phloem would be, around here. It's a very bad diagram. But where would transport of material be located? It will be inside this little thing here. So it'll be B. All this here would be like the cortex, and here outside here would be like the pericycle, but inside here is where you have a vascular bundle. Alright, we did 12 already. Endocytosis, the engulfing of a bacterium by a phagocyte. Bobby is given a fresh, right? Places in seawater and it dies. This is because water diffuses out of the fish. Yeah. We bounced this one. We bounced these two up already. Which of the following statements is correct about the rate of reaction between X and Y? You see X here, can't really fit with Y, so it have no. If we ignore induced fit, there'll be no reaction. And 15, the decline in the rate at X is the enzyme loses its tertiary structure. The base represented by X is, you remember this one? I think we only bounced this up once before. It was 16. Yeah. So it would be cytosine. Level type of explanation, right? Again. Right? Alright. 17. Which of the following features are true for chromatins but not for chromosomes? Okay. Well, Paul, what do you think? Tricky one. Yeah. Says all. Chromatins have loosely coiled long threads, so that's true, right? They do contain. Hold on. Which of the following features are true for chromatins but not for chromosomes? Chromosomes have histone. So 3 is definitely in, right? Because they have loosely coiled long threads. But not for chromosomes. chromatins would have the protein, but chromosome. Let me make sure. Let me look this up. Chromosome histones. I believe they do. Yeah, because chromosomes have the DNA packaged into histones. The chromatins don't have the chromatins are loosely, they don't have the proteins. Chromosomes have the proteins. So is this is no but wait. How but both of them contain DNA though. Chromatin, hold on. There's a real chain up one. But let's make sure they. No, but chromatin has histones. Chromatin has histones. The DNA wraps around the chromatin and has histones. Something wrong there. No, chromatin has. It does. It could. Look it up and make sure. Look it up and make sure. They have histones. Which are true for chromatins, but not for chromosomes. This is what chain them up here. Alright, something wrong with this question. Alright. I think it's supposed to be. No, well, it's how they word it. The chromosomes have DNA. That don't make sense for it to be all three. Chromosomes also are proteins. I think if they did say which of the following features are true for chromatins and chromosomes. Alright, leave this one out. Something wrong. Something wrong with that. Something off. Ask all the teachers. Something wrong with that question. Which of the following statements about semi-conservative replication of DNA is most appropriate? Um, okay, so we did this one already. Replicated DNA has one parental strand and one new strand. So one old and one new. All your accents, I don't know. Something wrong with that one. We do this one too. Which of the following combinations correctly identifies the stages 1, 2, and 3? So remember 1, the meat in the middle, so that's metaphase. 2, the disalignment, that's prophase. And 3, the angry, so it's anaphase. So 19 is C. We do this one already. Okay. 80, 20. Which of the following, this is real blurry. Which of the following occurrences is not characteristic of meiosis? We do this one too. It'll be A. Meiosis doesn't form identical sets of chromosomes. It forms four daughter nuclei with genetically varied chromosomes. 20 is A. 21. In a rat cell, there are 32 chromatids present at the beginning of the process of mitosis. How many chromatids would there be in the beginning of the cell of prophase 1? We do this one too. So this one here is the same. At the beginning of meiosis, it would have 32, but at the end, it would have 16. Right? So the beginning will have 32. The end of meiosis would have 16. All right. A null hypothesis is best described as a hypothesis. So remember this one. So the null hypothesis is where they say that there's no significant difference between the observed and expected data. Right? So the initial thing is to remember what you would do is you would accept, sorry, you would reject, or you would fail to reject. And that would be an alternative hypothesis. All right. Well, we did this one the last time, but it have a mistake, right? Does the one that have the mistake, eh? Because if you have ATA, this will be UAU. Oh no, they fix it. So UAU is tyrosine. TAT, this would be AUA, and that would be wait. No. It's not TAT. What shipping is I doing? Is GTA. Yeah, so GTA would be CAU. And CAU is histidine. Yeah. So yeah, they get correct this time. So there's the only one here that matches. Which of the following is true of restriction enzymes? We did this one, so it's A. They cut DNA at specific base sequences. They don't always produce sticky ends, right? So that's wrong. That's correct. Some of them don't. They should, but they don't. We did this one just now. Despite the benefits of gene therapy, many people have reservations about its use. Which are the following concerns about gene therapy may be considered a hazard? ABC. Because they use viruses as vectors, those could cause diseases in immunocompromised people. We did this one. A possible benefit of producing transgenic plants is improved human nutrition. The phenotype of an organism is best explained as those physical characteristics that do what? All right. Paul, you feel for that one? That is not C, you know. Yes, D. Yeah, that reflect the interaction of the genetic makeup and environment of the organism. So, for example, if I dark skinned, that is reflective of how much melanin is produced in my body, but is also reflective of the type of climate that I live in, how much times I go in the sun, and so on. So it's D. Alright, we did this one. Which of the following forms of gene mutation will result in a frame shift mutation? So basically, you're looking to see if you have an addition or deletion, because those could you know they could basically shift the reading frame of it. Aaron, you free to sleep in? You go and sleep. You waiting for me? So it's 1 and 3, but not an inversion. An inversion and a substitution do either add or take away a nucleotide. So is B. Aaron doing like he paying for the YouTube live, yes? Man could jump in and jump out whenever your mother have you here, or there are various isolated mechanisms which can result in population of the same species evolving into two distinct species. Which of the following is an example of behavioral isolation? We now do this. So is D. In species of, well, this first one here, this is temporal because they bloom in different times. This is ecological because they grow in two different types of environments. The flower the flower structure of one prevents the transfer of pollen. That's mechanical. And this one here, funnel web spiders, only males will spray the female with a chemical, right? So there's this behavioral. So it's a courtship behavior. All right, so we had this giraffe question already. And basically, if you had to draw this as a graph, you'd have your long neck ones over here, and your short neck ones, you barely have them. So when you have something like this is directional selection. I prefer one to the other one. All right, we favorite question. I feel like this module 3 does always start with this question. But anyway, this is correct and this is wrong. All right, so the one is D. Hibiscus and flamboyant use pollen, not spores, and they're not asexual. All right, so 32. Which of the following statements describes the events occurring in asexual reproduction only? All right, so that's A. We did this one early. Offspring are genetically the same and gametes are never involved. The most important advantage of sexual reproduction is that it allows adaptation. We did this one already. To change in environmental conditions, natural selection, and survival of the fittest, right? All right, next favorite one. The ovary is best described as a swollen base of a single carpel. All right, so we bounced this one up already. So remember here, this is meiosis that's producing these four cells here, these four megaspore cells, and this surviving megaspore is going to undergo growth, right? Then mitosis, 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 and then fusion. Basically, these two polar nuclei are going to fuse to form a central cell. And yeah, which one is that? Yeah, so C. Androsium matures before gynosium. That's protandry or protandrous. Okay, we bounce up a unique one here. The structure labeled Y. Anybody know what it's called this thing? Here we have an anther. And this entire thing is a pollen sac. So what is this layer that is around, well, this inner layer in the pollen sac here? Nah, it's not the epidermis. Yeah, so it's a tapetum. 35 is A? Oh shucks. Sorry. 35 is A. 35 is A. 35 is A. Hard luck day. 34 is C. 35 is A. Tapetum. Tapetum is actually a Latin word for carpet. Don't know why I call it that. But yeah. But it basically provided nutrition for the pollen cells in there. Yeah, I looking to wrap up. 38. Which of the following, we just do this, occurring is a result of the negative feedback caused by an increase in estrogen levels. Right? So negative feedback, increase in estrogen leads to decrease in FSH. Now bounce this up to which of the following is the difference between a secondary oocyte and the ovum? Secondary oocyte is at metaphase 2. Ovum has completed all meiotic divisions. Now bounced up. 41. Which of the following cells are the nutritive cells of the seminiferous tubules? Give me an answer there for 41. Yeah. So there's your Sertoli cells. Your Sertoli cells. Your Sertoli cells is basically your nurse cells. That's what's gonna feed the spermatids, and eventually they will develop into spermatozoa. In human in human males, the paired testes are external to the body cavity. All right. Well, boys know this. The external because they're too big to be. That's not. That's not. This is so that they contain sperms which need a cooler temperature to remain viable. The So when you go in the shower and the water real cool, and the testicles it going right up. Well, that's because it cold outside and it's time to get some warmth. So it reaching back inside to reach your normal body temperature. I mean, yeah. So you get a thing called shrinkage that happening now because it's trying to warm up. It's trying to run away from the cold. That's basically what happening. The thing like it's smart. All right. The importance of the amnion include what? Okay, so the amnion is what contains the amniotic fluid. All right, so answer for 43 then. All right, so yeah, so it's all. The amnion protects the developing fetus from mechanical shock. Basically, the pregnant woman get a lash in she belly, the baby is bound to get the lash too. Genetic screening via amniocentesis. You could basically tell, you could get a small sample of the amniotic fluid and you could actually get genetic material from it. See if the baby, there's something wrong. Provision of a medium for proper fetal limb development because the baby constantly moving and there's resistance with the fluid. Our next thing is the amnion, because a lot of the amniotic fluid is water and water has a high specific heat capacity, is that it helps with regulation of body temperature, you know, prevent the baby from getting too hot, you know what I mean? Alright, so 44. Which of the following is an important function in the mammalian placenta? Is A, secretes hormones like progesterone, HPL, and HCG. Which of the following best explain? Hey, real bongs. There's like I feel like there's a classic last question, right? Anyway, so it stops it from developing spinal bifida because it helps develop the neural spine. All right, so I'm gonna link up something to all you here. Um, Before I go. But you have to make sure and get some sleep. Don't drink no milk and eat no curry in the morning. You know what I mean? Before you have to go. Alright. So all you can see our file there. Alright, so okay, so not right. So all you can see there. You're feeling better for tomorrow. That's good. Well, I say I come on here. I tell you earlier. I here to ease up our stress. So all you go pay it forward to somebody later on. if somebody need help or they help them out. Wait, well, let me show you something they um hold on, eh? I'm trying to get a little thing there. Where am I Chrome boy? Alright. Okay, so if all you watch here, I basically compile a bunch of the multiple choice solutions here. So those who wanted I don't do bio lessons, but I is not my teacher. I is not my teacher, son. I is not my teacher, so don't even ask for lessons. Alright, so the check out all the solutions there. And the paper two, well, what I gonna do by I don't know how they're gonna. Well, all they could me you know, I go post a little thing for the YouTube channel tomorrow, but I don't know what I'm doing tomorrow. I feel I have to go vaccinate tomorrow, vaccinate to operate, all you know what I mean. Nah, I'm not wrong for unit two. Well, actually, I might be, but like I might do a revision thing next year for it, but you can't count on that, you know what I mean? I'm busy. It's only right now I'm not busy. Like for the next for this month, me I'm busy, but I might be busy from then. Yes, so don't ask me for no lessons and thing because again, I was not a teacher. Okay, so that is it. Well, I'll take care, and tomorrow we go vibes again. I really don't know. I don't know how I get this link because literally I give this link to like two people and I post it on my Facebook. So I assume the link just pass around. So how I get the link? I don't know. Or I could subscribe to the channel. The channel doesn't have no videos on it. Right. Well, I go post a link tomorrow too, but I could just keep the channel there. And I go put up a schedule thing tomorrow. Alright. Well, yeah. How you just save this thing? I don't know how to use YouTube. Let me see how you do this. I don't know who I'm watching this. Um, we are streaming for a real long way. Alright. Well, that's the deal. Yeah. Laters.